morning dear students. In today's class we shall study about the hormones of adrenal medulla. As you all know, adrenal gland is a small triangular shaped gland present over the kidneys. Yes, it is a small gland present over the upper pole of the kidney. This adrenal gland has two parts. One is the adrenal cortex, the outer part what we call it as adrenal cortex. The inner part is called as adrenal medulla. This adrenal cortex secretes various enzymes like various hormones, that is the cortisol, glucocorticoids, aldosterone, sex steroids, all these are secreted from adrenal cortex. Whereas adrenal medulla, that is the inner part of the adrenal gland, secretes certain hormones, mainly the catecholamines. We call these hormones as catecholamines. The catecholamines are the main important catecholamines secreted from adrenal medulla are epinephrine, norepinephrine and dopamine. Now let us see all of them in detail. Adrenal medulla is formed by two types of cells histologically. These cells are called as chromaffin cells. Chromaffin cells are also called as pheochrome cells or chromophil cells. They are also called as chromophil cells. They take up the coloring agents in the stain. So we call it as chromophil cells or pheochrome cells. The adrenal medullary hormones, mainly the adrenaline secreting cells, are more in number. About 90% of the chromophil cells secrete adrenaline, whereas about 10% of the cells in the adrenal medulla secretes noradrenaline. Very few cells secrete dopamine. Noradrenaline and dopamine act more as neurotransmitters. They are released from the nerve terminals more than the adrenal gland. The main secretion of adrenal gland is adrenaline. It is also called as epinephrine. We use the terms as synonyms: adrenaline and epinephrine, noradrenaline and norepinephrine. These two terminologies are used like synonyms. Okay, don't get confused with these terms. Adrenal medullary hormones are mostly amines. Structurally, they are amines. They are derived from catechol, and so these hormones are called as catecholamines. Structurally, they are amines derived from catechol, so we call them as catecholamines. So let us see what are these catecholamines. Catecholamines are the hormones secreted by adrenal medulla. Catecholamines include adrenaline or epinephrine, noradrenaline or norepinephrine, dopamine, and other types of catecholamines include dynorphins, neurotensin, substance P, enkephalin and somatostatin. These are other types of catecholamines. These substances may be released in other structures also. About 80% of adrenal medullary catecholamine is epinephrine. We have 90% of the chromophil cells secreting adrenaline, and the rest is norepinephrine and few other hormones. Epinephrine circulating in blood is almost exclusively produced in adrenal medulla, whereas norepinephrine, the dopamine, other catecholamines, they are also secreted from other structures in the body, whereas epinephrine or adrenaline is totally secreted from adrenal medulla. Norepinephrine is secreted by adrenal medulla in small amounts, but it is secreted at the nerve terminals as a neurotransmitter. It is a neurotransmitter, but in select circumstances it may also function like a hormone. The concentration of epinephrine, norepinephrine is highest in hypothalamus. Norepinephrine is its concentration is highest in hypothalamus. Coming to the plasma levels of catecholamines, adrenaline is about 3 micrograms, adrenaline is about 3 micrograms per dl in blood. Noradrenaline or norepinephrine is about 30 micrograms per dl, and dopamine is around 3.5 micrograms per dl. And its half-life is very short. The duration is very short, about 3 to 5 minutes. On an average, the catecholamines stay in the plasma for about 2 minutes. On an average, they stay in the plasma for about 2 minutes. So it is very difficult to do an assay of these catecholamines. So in order to find out what is the amount that is being secreted, what is the amount present in plasma, it is very difficult, but it's because its half-life is very short. 3 to 5 minutes, on an average it is just 2 minutes. These catecholamines are mainly synthesized from tyrosine. If you remember, tyrosine acts as a parent hormone or parent substrate for the synthesis of thyroid hormones as well as catecholamines. The tyrosine is a parent amino acid for the synthesis of either thyroid hormones or the catecholamines. Now let us see how these catecholamines are synthesized. The stages of synthesis of catecholamines. Formation of tyrosine is the first step in the synthesis of catecholamines. This occurs anywhere in the body. Tyrosine is synthesized. The formation of tyrosine from phenylalanine requires the enzyme called as phenylalanine hydroxylase. It requires the enzyme phenylalanine hydroxylase. With the help of this enzyme, phenylalanine hydroxylase, tyrosine will be formed from phenylalanine. So this tyrosine is synthesized in either in liver or in other parts of the body. Once it's synthesized, this tyrosine is taken up from the adrenal medulla. So the actual substrate for synthesis of catecholamine is tyrosine. Tyrosine is synthesized from phenylalanine. Okay. So this tyrosine is taken up from the blood into the chromophene cells in the adrenal medulla by active transport mechanism. Once tyrosine is taken up by the chromophene cells in the adrenal medulla, now synthesis of catecholamine begins in the adrenal medulla with the substrate tyrosine. So remember, tyrosine is the first amino acid or the substrate amino acid which is required for synthesis of catecholamines. Now tyrosine is converted to DOPA, that is dihydroxyphenylalanine. Dihydroxyphenylalanine, we also call it as DOPA, T-O-P-A. Tyrosine is converted to DOPA by the enzyme, by the presence of the enzyme tyrosine hydroxylase. Tyrosine undergoes hydroxylation with the help of the enzyme tyrosine hydroxylase to form DOPA, dihydroxyphenylalanine. Right? So this is the first step in the synthesis of catecholamines. Tyrosine is converted to DOPA with the help of the enzyme tyrosine hydroxylase. Yes, tyrosine is converted to DOPA with the help of enzyme tyrosine hydroxylase. This occurs in the cytoplasm of the chromophile cells inside the adrenal medulla. Now DOPA is converted to dopamine with the help of the enzyme DOPA decarboxylase. It undergoes decarboxylation. Dihydroxyphenylalanine undergoes decarboxylation to form dopamine with the help of an enzyme DOPA decarboxylase. Again, this occurs inside the cytoplasm. The dopamine so formed will be taken up into the granules. It will be taken up into the granules inside the chromophil cells. Dopamine is further converted to norepinephrine by dopamine beta hydroxylase, which is present inside the granules of the cell. Dopamine beta hydroxylase converts dopamine to norepinephrine. So first amino acid that is required for the synthesis of the catecholamines is the tyrosine. Tyrosine is converted to dopa. Dopa is converted to dopamine. Dopamine is converted to norepinephrine. From norepinephrine, the phenylethanolamine N-methyl transferase converts norepinephrine to epinephrine. Again, epinephrine is also taken up by the granules. Norepinephrine so formed inside the granules, they diffuse into the cytoplasm. In the cytoplasm, norepinephrine is converted to epinephrine. Epinephrine so formed can be again taken up into the granules. So this is how the catecholamines are synthesized: dopa, norepinephrine and epinephrine. To summarize, phenylalanine is converted to tyrosine in the blood by phenylalanine hydroxylase. So the amino acid which acts as a substrate for the synthesis of catecholamines is present in the blood, that is the tyrosine. Tyrosine is taken up by the chromophil cells. This tyrosine is taken up by active transport mechanism. Tyrosine is converted to dopa by tyrosine hydroxylase. Hydroxylation occurs and it forms dihydroxyphenylalanine. Dihydroxyphenylalanine is converted to dopamine by dopa decarboxylase. Here it undergoes decarboxylation. Now the dopamine so formed is taken up into the granules. Inside the granules, dopamine is converted to noradrenaline or norepinephrine. Both are same. Norepinephrine or noradrenaline is formed by dopamine beta hydroxylase. Hydroxylation occurs and with the help of beta hydroxylase, dopamine is converted to noradrenaline. Noradrenaline diffuses out of the granules, outside the granules with the help of the enzyme PNMT. It forms adrenaline. Adrenaline moves into the granules. So this is how the synthesis of catecholamines occur in the adrenal medullary cells. Now these catecholamines so synthesized are stored in the form of granules in the chromophil cells. It is taken up by the chromophil granules and stored inside the granules. So this is about the synthesis of the catecholamines. Once these catecholamines are synthesized, they circulate all over the body, then they are going to be degraded. It is getting degraded into form. Adrenaline forms meta-adrenaline and then vinyl mandelic acid. Noradrenaline or norepinephrine forms meta-noradrenaline and vinyl mandelic acid. There occurs methoxylation and oxidation of these enzymes and they are going to be degraded in the body. Now we shall see that in the metabolism. So this is about the synthesis of the various catecholamines. Now once these catecholamines are synthesized, they are secreted in the ratio of 4:1. Secreted epinephrine and norepinephrine in the adrenal medulla is in the ratio of 4:1. That is, more and more epinephrine or adrenaline is synthesized than the norepinephrine. Basal plasma levels of free epinephrine are about 30 picograms per ml and that of free norepinephrine is higher. It is 300 picograms per ml because norepinephrine, other than adrenal medulla, it is mainly synthesized in the nerve terminals. So its concentration is more compared to epinephrine. Dopamine is present in minor quantities. It is about 35 picograms per ml. It is mainly seen again as a neurotransmitter in the brain. The plasma half-life of these hormones, epinephrine, norepinephrine, is extremely short. As we know, the average plasma half-life is just 2 minutes. It ranges from 1 to 3 minutes or 1 to 5 minutes. About 30 micrograms of norepinephrine and 6 micrograms of epinephrine and 700 micrograms of vinyl mandelic acid are excreted in the urine per day. This much amount is going to be excreted in the urine per day. Vinyl mandelic acid is a metabolite formed when epinephrine and norepinephrine are degraded, right? So this is about the circulating levels and excretory levels of these hormones. As we know, the plasma levels of these hormones is also minimal and the half-life is very short. It is very difficult to do the assay of these hormones, the plasma levels of these hormones. So what we do is we indirectly assess the concentration of these hormones that is getting excreted. So if the concentration of the hormones that is getting excreted is in the normal levels, we consider the secretion of hormones in the plasma is also under normal levels. If the concentration of excretion is less or high compared to the normal levels, then that is being reflected on the production level also. If it is lesser, the excretion level is lesser, then synthesis of these hormones is considered to be less. If excretion of these hormones is more, then we consider the synthesis of catecholamines is increased. So this is how we do the assay of these hormones. Yes. About 85% of noradrenaline is taken up by the sympathetic adrenergic neurons. The remaining 15% of noradrenaline and adrenaline are actually degraded. How they are degraded? The steps in the metabolic disposition of catecholamines. Norepinephrine is converted into nor-metaepinephrine with the help of the enzyme COMT. Normetanephrine is converted to methyl hydroxyphenyl glycol, MOPG. Now, epinephrine is converted to metanephrine. It is also converted by MAO enzymes into dihydroxymandelic acid and then into vinyl mandelic acid. So we actually do the assay of vinyl mandelic acid to know the which indirectly reflects the synthesis of these catecholamine hormones, norepinephrine and epinephrine. So these are the different metabolites of norepinephrine and epinephrine. Normetanephrine, dihydroxymandelic acid, metanephrine and vinyl mandelic acid. Finally, it is converted to methoxyhydroxyphenyl glycol. Methoxyhydroxyphenyl glycol is again converted to vinyl mandelic acid. So the final substrate which is being excreted is in the form of vinyl mandelic acid. So this is about the steps in metabolic disposition or degradation and excretion of these catecholamines. So removal of catecholamines, they are removed from the body through urine in 3 forms. 15% of free adrenaline and free noradrenaline is being excreted. 50% of free or conjugated meta-adrenaline or meta-epinephrine and meta-norepinephrine are excreted. 35% is excreted as vinyl mandelic acid. Why we are stressing so much on these metabolites? Because we are also assessing how much of these metabolites are excreted in order to know how much the synthesis of catecholamines is going to take place. Whereas we are not bothered about excretion of other hormones because we can easily do the assay of those hormone levels in the plasma itself. Since we cannot assess catecholamine secretion in the plasma, or it is very difficult, we are also concerned about the metabolites which are going to be excreted, right? So this is about the removal of catecholamines. Now coming to the mechanism of action and functions of catecholamines. Remember, catecholamines, that is adrenaline or epinephrine, noradrenaline or norepinephrine, they act on two types of receptors. They act on two types of receptor. One is the alpha receptor. One type of receptor is alpha receptor. Alpha receptor again has two types, alpha 1 and alpha 2 receptor. Beta receptors is another type. Beta receptors again have three subdivisions or three subtypes: beta 1, beta 2 and beta 3 type of receptors. So we have two types of receptors on which adrenaline or noradrenaline is going to act: alpha and beta receptors. Alpha receptors are again subdivided into alpha 1 and alpha 2. Beta receptors are subdivided into beta 1, beta 2 and beta 3. Now let us see where these receptors are present. What is the mechanism through which the hormones act through these receptors? Where are these hormones situated? When the hormone acts on this receptor, what is the function that is going to be subserved? So let us see all of them in detail. The catecholamine hormones, adrenaline or noradrenaline, acts through alpha receptor, that is alpha 1 receptor, with the mechanism of activating phospholipase C. I repeat, it activates IP3 DAG mechanism through phospholipase C, activation of phospholipase C. When the hormone attaches to the receptor present on the cell membrane, all these receptors are cell membrane receptors. Hormone acts on the receptor, noradrenaline combining with the receptors present on the plasma membrane. They are going to activate the phospholipase C. Phospholipase C in turn converts to form IP3 and DAG. Phosphatidyl inositol is converted to phosphatidyl triphosphatidyl inositol and DAG, diazyl glycerol. These two in turn is now going to activate certain calcium channels. It is going to release the calcium and also it is going to synthesize more and more hormones or receptors required for its action. It is mainly going to increase the calcium levels inside the cells and it is going to perform its function. Right? So this is about the mechanism of action of alpha 1 receptor. I repeat, on alpha 1 receptors, combination of hormone on the receptor activates phospholipase C, which in turn activates the second messenger system, that is IP3 and DAG, which is going to increase the serum calcium level. Increasing serum calcium level will further perform the functions which is going to happen because of this. Right? So this is the mechanism of action of hormone on alpha 1 receptor. Now coming to the other receptor, mechanism of action through other receptor. The epinephrine or norepinephrine attaching to the receptor present on the cell membrane. This receptor, if it is alpha 2 receptor, this alpha 2 receptor is going to activate inhibitory G proteins. G proteins, you have heard of G proteins, right? So these G proteins are of several types. It could be stimulatory or it could be inhibitory. Here, if it is alpha 2 receptor, it is going to inhibit the G protein coupled receptor. So with this inhibition, it is going to inhibit the conversion of GTP to GDP. It is also going to inhibit the conversion of ATP to cyclic AMP. So it is mainly inhibitory in function. So the hormone acting on alpha 2 receptor, mainly it is going to inhibit. inhibit the several cascade of mechanisms. Now we have other type of receptor, that is beta receptor. Beta receptor, if the hormone combines with beta receptor, then beta receptor is going to activate stimulatory G proteins. That is, it is going to stimulate the G protein coupled receptor. So this stimulation of G proteins is now going to convert activate adenyl cyclase, which in turn converts ATP to cyclic AMP. Cyclic AMP is going to activate protein kinase A, phosphorylate the proteins, and physiological function is subserved. So cyclic AMP pathway will be activated. The second messenger system here is the cyclic AMP. So hormone acting on alpha 2 receptor, it is going to inhibit the G proteins. Hormone acting on beta receptors, it is going to activate the G protein. It is going to convert ATP to cyclic AMP and then activates the protein kinase A. Activation of several enzymes and further mechanism of action. The functions of these hormones will be performed. So like this, the catecholamines, though the hormone is same, depending upon the receptor on which it is going to act, the functions of the hormone is going to be different. Now let us see what are the functions depending upon the receptor that is going to be activated. Hormone acting on alpha 1 receptor acts on smooth muscle. It causes contraction of the smooth muscles. Contraction of smooth muscle in the blood vessel, eyes, bladder, prostate and uterus. I repeat, the hormone acting on alpha 1 receptor, catecholamine hormones, adrenaline or noradrenaline, especially it is noradrenaline. Noradrenaline mainly acts on alpha 1 receptor. It is going to increase the smooth muscle contraction in the blood vessel. What happens if the smooth muscles in the blood vessel contract? The lumen of the blood vessel is going to reduce, right? Lumen will reduce. If the lumen of blood vessel reduces, what happens to blood pressure? Blood pressure should increase. Peripheral resistance increases and the diastolic blood pressure has to increase, right? Then in the eyes it causes constriction of the pupil. Bladder, contraction of the bladder, more so it causes contraction of the sphincters. Prostate, it causes contraction of smooth muscles, contraction of smooth muscles in the uterus. So this is the action of alpha 1 receptors on smooth muscle. Now action of alpha 1 receptors on liver, it increases the metabolism of glucose. What it does is, it causes glycogenolysis. It lyses the storage form of glucose, that is glycogen. So glycogen is lysed and more glucose will be released into the blood. It leads to a hyperglycemic state, right? So this more and more glucose is used as an instant energy whenever it is required for activation. For example, these hormones, the adrenal hormones, are more for fight and flight reaction. Whenever an emergency arises, for immediate functioning we require these adrenal hormones. So these hormones increases the plasma glucose level, which acts as a instant energy. So it causes glycogenolysis. Storage form is lysed and plasma glucose will be increased. In salivary glands, it increases the secretions in the salivary gland. At GI, that is in the gastrointestinal tract, the alpha 1 receptors causes relaxation of the smooth muscle instead of contraction. It is ulta, that is, it is opposite to that of contraction in the other muscles. In the GI tract, through the alpha 1 receptors itself, it causes smooth muscle relaxation instead of contraction. So this is about the function of adrenaline and noradrenaline, more of noradrenaline on alpha 1 receptor. Yes. On the alpha receptors, which is present in various locations, on the alpha 2 receptors. Alpha 2 receptors mainly present on the presynaptic surface of the neurons. Presynaptic neurons. On presynaptic neurons, the hormone acts through the alpha 2 receptors. It causes decrease in noradrenaline release. It decreases the acetylcholine release. It also decreases the insulin release. So by decreasing all of them, noradrenaline, acetylcholine and insulin, it is diverting all of the functions for fight and flight reaction. So noradrenaline release is decreased, acetylcholine release is decreased and insulin release is also decreased. Alpha 2 receptor, I told you, it acts by inhibiting the G protein. So it is mainly a inhibitory hormone. The hormone will act as a inhibitory hormone when it acts through alpha 2 receptor. So it is going to inhibit the release of various hormones. So this is about the action of hormones on alpha 1 receptor and alpha 2 receptor, right? So we shall move further. So no confusions. The same hormone acting on different receptors, it is going to perform different functions. On alpha 1 receptor, it is acting on the smooth muscles, causing smooth muscle contraction except in the GI tract where it causes relaxation. In the liver, it is causing glycogenolysis, increasing the glucose concentration in the plasma, and it is also going to cause lipolysis. We will see all of them. On alpha 2 receptor, it is acting as inhibitory in function, decreases the release of noradrenaline, acetylcholine and insulin. So this is about action of the hormones on alpha receptors. Now action of hormones on beta receptors. On beta 1 receptor, if the hormone is acting on beta 1 receptors, beta 1 receptors are mainly present on the heart, on the cardiac muscle, and it is the adrenaline or epinephrine which is going to act mainly through beta receptors. Noradrenaline acts mainly through alpha receptors. Adrenaline or epinephrine acts mainly through beta receptors. It is not mandatory. Both the hormones may act on all types of receptors, but most of them acting on these receptors. Okay. Beta 1 receptors, action of epinephrine or adrenaline on beta 1 receptors, it is going to stimulate the heart. It is going to increase the rate of heart, that is heart rate increases, force of contraction increases, and it also increases the automaticity. It increases the excitability in the cardiac muscles. Beta 2 receptors are mainly present on the smooth muscles. The hormone acting on beta 2 receptors is going to cause smooth muscle relaxation. I repeat, the hormone epinephrine or norepinephrine acting on beta 2 receptors is going to cause relaxation of the smooth muscles. Now beta 3 receptors are mainly present on the adipose tissue. It is going to increase lipolysis. Catecholamines will increase the lipolysis by acting on beta 3 receptors. So this is the gist of the hormone action on various receptors and the functions subserved. Now let us see the functions of catecholamines in detail. Functions of catecholamines on metabolism, acting through both alpha as well as beta or either alpha or beta receptor. On metabolism, adrenaline influences the metabolic functions more than the noradrenaline. So it is adrenaline which is going to influence the metabolic function rather than noradrenaline. Remember, it is a calorigenic hormone. Calorigenic hormone means it is going to increase the oxygen consumption, it is going to increase the basal metabolic rate. So it is a calorigenic hormone. Adrenaline increases oxygen consumption and carbon dioxide removal. It increases the basal metabolic rate. On carbohydrate metabolism, it is a hormone which causes increase in plasma glucose level. So increase in plasma glucose level by lysing the storage form or by increasing gluconeogenesis. So adrenaline increases the blood glucose level by glycogenolysis in liver and muscle. Then, on fat metabolism, adrenaline causes mobilization of fatty acids from adipose tissue. Catecholamines need the presence of glucocorticoids for this action. So glucocorticoids has a permissive role on the adrenal medullary hormones. If glucocorticoids are present, then it leads to this function. Adrenaline causes mobilization of free fatty acids from the adipose tissue. On the blood, via beta receptors, adrenaline decreases the blood coagulation time. It also increases RBC count by contracting the smooth muscles of splenic capsule. So whatever is present in the spleen, the sinusoids of the spleen will be squeezed and released into the circulation. Action of adrenaline on heart. This action is via the beta receptors, especially beta 1 receptor. Adrenaline has a stronger effect on heart than the noradrenaline. It increases the overall activity of the heart. Remember the fight and flight reaction. Whenever an emergency situation occurs, you want your heart to pump more, your muscle to act, so that you will gain more and more blood supply to the muscle. Your heart pumps more blood, your blood supply to skeletal muscle is more, and you can run away or you can escape from this dangerous situation, right? So for this we need increase in cardiac activity. What all increases? Heart rate increases. This is called as chronotrophic effect. Force of contraction of cardiac muscle increases. This is called ionotrophic effect. There is increase in excitability of the heart muscles. There is easily excitation of SA node, which leads to bathmotrophic effect. And conductivity in the heart muscle is also increased. We call it as dromotrophic effect. So these are the effects of adrenaline and noradrenaline on heart via beta receptors. It is more of adrenaline which is going to act on beta 1 receptor rather than noradrenaline. Now, what is its function on blood vessels? On blood vessels, via the alpha receptors, noradrenaline has a stronger effect compared to adrenaline. So this noradrenaline is going to cause vasoconstriction, because noradrenaline acting on alpha 1 receptor as a smooth muscle causes smooth muscle contraction, isn't it? So it causes vasoconstriction. It causes generalized vasoconstriction. It is a general vasoconstrictor. So this vasoconstrictor effect of noradrenaline increases the total peripheral resistance. So alpha 1 receptor, hormone acting through alpha receptor is going to cause vasoconstriction. Whereas the hormone acting on beta 2 receptors, adrenaline acts on beta 2 receptors more than noradrenaline. So it is mainly adrenaline which is going to act through beta 2 receptor present on the blood vessel. So this adrenaline causes vasodilation more so in the blood vessels present in the skeletal muscle, liver and heart. I repeat, the adrenaline acts through beta receptors which are present on the blood vessels, especially in the structures like skeletal muscle, liver and heart. It causes vasodilation. Here it causes vasodilation. Adrenaline acting through beta 2 receptors present on the blood vessels in the skeletal muscle or in the liver causes vasodilation. Whereas noradrenaline acts acting through alpha receptors causes vasoconstriction. Adrenaline acting through beta 2 receptors causes vasodilation. When vasodilatation occurs, there is decrease in the total peripheral resistance. So this is about the function of the adrenaline and noradrenaline on blood vessels. For the action of adrenaline or noradrenaline on the blood vessels, catecholamines needs the presence of glucocorticoids. Again, it is a permissive action of glucocorticoids on the catecholamines. On blood pressure, what happens to blood pressure because of these hormones? Norepinephrine increases the systolic blood pressure because it increases the force of contraction of the heart. At the same time, norepinephrine is going to cause vasoconstriction in the blood vessels, so it causes increase in total peripheral resistance. Increase in total peripheral resistance increases the diastolic blood pressure, right? So norepinephrine is going to increase both systolic blood pressure is also increased, diastolic blood pressure is also increased. Whereas action of epinephrine acting on heart has 4 effects: increase in chronotrophic, dromotrophic, bathmotrophic, all inotropic, all of them is increased, right? So it increases the systolic blood pressure because it is increasing both heart rate and force of contraction, increase in systolic blood pressure. But epinephrine acting on blood vessels, it is causing vasodilation. So vasodilatation leads to decrease in the peripheral resistance. So there is decrease in the diastolic blood pressure. So norepinephrine is increasing both systolic and diastolic blood pressure. Epinephrine is increasing systolic blood pressure, but it is decreasing the diastolic blood pressure. So pulse pressure becomes wide. Pulse pressure means systolic blood pressure minus diastolic blood pressure is called as pulse pressure. So pulse pressure becomes wide with the action of epinephrine, right? So this is about the functions of catecholamines on blood pressure, or what is its effect on blood pressure. Now what is the effect of catecholamines on respiration? Via beta 2 receptors, adrenaline is going to increase the rate and depth of respiration. Adrenaline is increasing both rate and depth of respiration. Adrenaline injection will produce an apnea, that is stoppage of respiration momentarily because of increase in rate and depth. More and more oxygen is accumulated, carbon dioxide becomes very less, carbon dioxide washout occurs, so stimulation of chemoreceptors does not occur. This leads to adrenaline apnea. This is called, at the same time, adrenaline also causes bronchodilation. So this is the effect of adrenaline on respiration. Now the effect of adrenaline on skin via both alpha and beta 2 receptor. Adrenaline causes contraction of erector pilate, so we call what we call it as goosebumps. All the hairs will stand on the skin. It also increases the secretion of sweat through the apocrine glands. The action of these catecholamines on skeletal muscle. Via alpha as well as beta 2 receptors, adrenaline causes severe contraction of skeletal muscle and it causes easy fatigue of skeletal muscle. But this severe contraction is required to escape from the dangerous situation in the case of fight and flight reaction. It also increases glycogenolysis and the release of glucose from muscle into the blood. It also causes vasodilatation in skeletal muscle. So by the action of adrenaline through beta 2 receptor, there is vasodilatation in the skeletal muscle, right? So this is about the function of adrenaline on skeletal muscle. It increases the contraction of the skeletal muscle. It also increases the blood supply to the skeletal muscle It increases glucose supply to the skeletal muscle by increasing the plasma glucose level. So this is about the, but at the same time the glycogenolysis occurs both from liver as well as from the muscle. So this is about the effect or function of catecholamines on skeletal muscle. Coming to smooth muscle. Contraction of following smooth muscle occurs. Splenic capsule, hence more and more stored blood is going to be released into circulation. Sphincters of GI tract, there is contraction. Erector pili of skin, there is contraction of erector pili which leads to goosebumps. Contraction of gallbladder, uterus, there is contraction of dilator pupillae of iris and mitigating membrane in case of cat. At the same time, there is relaxation of smooth muscles in certain organs. The non-sphincteric part of GI tract is relaxed. Esophagus, stomach and intestine, there is relaxation of the smooth muscles over these areas. Relaxation of the bronchioles, so there is bronchodilation, and urinary bladder. There is detrusor muscle relaxation but sphincter contraction occurs. So this is about the function of adrenaline on smooth muscle. Now action of catecholamines on central nervous system. Via the beta receptors, adrenaline is going to increase the activity of brain. So in case of an emergency situation, fight and flight reaction, the person has to act immediately and he has to think. Quick response has to be produced and he has to escape from the situation. So adrenaline increases the activity in the brain. Adrenaline secretion increases during the fight and flight reaction after exposure to certain stress. To come out of this stress factor, to think more how to come out of the stress factor, adrenaline is going to be increased. It also enhances the cortical arousal and other facilitatory functions of central nervous system. The other effects of catecholamines on salivary glands, it increases the salivary secretion. On sweat glands, it increases the sweat secretion by acting through the apocrine sweat glands. On lacrimal glands, it is going to increase the secretion of tears. It is also going to increase the ACTH secretion via alpha receptors. Adrenaline is going to increase the ACTH secretion. So these are action of the catecholamines on various secretions, salivary glands, sweat glands and lacrimal glands, and also secretion of ACTH. On nerve fibers, it is going to increase the conduction of the electrical activity. It is also going to increase the renin secretion by acting through beta receptors. This is about the functions of catecholamines on various structures. What are its functions? What are the functions through alpha receptors and through beta receptors? Now to summarize the functions, it causes bronchodilation, GI and urinary tracts, decreases the motility, decreases energy use, adipolysis occurs in liver, increase in glycogenolysis, increases gluconeogenesis and ketogenesis. In the skeletal muscle, increased glycogenolysis, and also it causes vasodilatation in skeletal muscle. Beta cells decrease in insulin secretion, alpha cells increase glucagon secretion. On the heart, it has a positive effect. It increases the heart rate, positive inotropic, chronotropic and dromotrophic effect. On the veins and blood vessels, there is vasoconstriction in all parts of the body, whereas vasodilatation in skeletal muscle and mainly liver. Then splanchnic artery involves vasoconstriction occurs. So by these, there is increase in venous return and increase in cardiac output. By the action on the lungs, there is increase in exchange of oxygen and carbon dioxide. So there is increase in blood supply, increase in oxygen supply. At the same time, by acting on the metabolism, there is increase in instant energy supply in the form of glucose. So all these are going to facilitate a fight and flight reaction response during a fight and flight reaction. Also action on brain, it increases the activity of the brain, it increases the conductivity, electrical conductivity in the nerves. So this is about the gist of functions of catecholamines. I hope you have understood the functions and no doubts regarding it. Functions of catecholamines is very important, both from clinical point of view and also from your exam point of view. Now, when these catecholamines are secreted more and more, as we have been telling, all these catecholamines are going to respond more when there is an emergency situation like fight and flight reaction. Apart from this, whenever there is a hypothermia, decrease in body temperature, decrease in external temperature, hypoglycemia, hypovolemia, during stress, various stress like trauma, anxiety, pain, all these conditions, there is increase in secretion of catecholamines. So these catecholamines prepare the body for any type of minor or major stressful situation, right? So these are the stimulus for secretion of catecholamines. Now coming to dopamine. Till now we have studied in detail about norepinephrine and epinephrine, or noradrenaline and adrenaline, which I repeat again and again. Norepinephrine, noradrenaline used as synonyms. Epinephrine and adrenaline are used as synonyms. Now coming to dopamine. Dopamine is again secreted by adrenal medulla, but it is mainly secreted from the dopaminergic neurons present in some areas in the brain, particularly in the basal ganglia. Injected dopamine produces the following effects. It causes vasoconstriction by releasing the norepinephrine, vasodilatation in the mesentery, increases heart rate, it increases the systolic blood pressure. So these are the effects of injected dopamine. Deficiency of dopamine, especially in the basal ganglia, leads to a neurological disorder called as Parkinsonism. So this is about dopamine in brief. Now, if there is abnormality in the secretion of catecholamines, the important cause for increase in secretion of catecholamines is tumor. Tumor of chromophil cells in adrenal medulla, we call it as pheochromocytoma. I repeat, tumor of chromophil cells in the adrenal medulla is called as pheochromocytoma. It is an endocrine disorder. This endocrine disorder leads to secondary hypertension. Why secondary hypertension? If you remember the functions of catecholamines on blood pressure, it is going to increase systolic blood pressure, and sometimes it is going to decrease diastolic blood pressure through beta receptors, increase diastolic blood pressure through alpha receptors, right? So these hormones are mainly going to cause increase in systolic blood pressure, so there is secondary hypertension. Symptoms include hypertension, headache due to hypertension, sweating, anxiety, palpitations because cardiac activity is increased more and more, chest pain occurs, and there is low orthostatic hypotension present. Orthostatic hypotension means when you lie down, you get up immediately, you get giddiness, we call it as orthostatic hypotension. To say orthostatic hypotension, blood pressure should reduce, systole should fall more than 20, diastole should fall more than 10. This probably could be because of vasodilatation in spleen, liver and skeletal muscle. That could be the reason for orthostatic hypotension. Though this condition has secondary hypertension which is not responding to drugs, it also shows orthostatic hypotension. So this is about the disorder of the adrenal medulla, that is pheochromocytoma. Tests for pheochromocytoma: measuring the metanephrines and vinyl mantelic acid levels in urine, and also catecholamine levels in plasma, which is very, very difficult. So this is about the catecholamines, how these catecholamines are synthesized, what are the functions of catecholamines, what are the receptors through which these catecholamines are going to function, and we also studied little bit about the dopamine and a gist of pheochromocytopenia. Any doubts you can put up in the group. Following roll numbers, if you are present, please post your attendance. Roll number 68, 102, 139 and 148. I repeat, roll number 68, 102, 139 and 148. If you are live, please put your attendance immediately. Immediately within 1 minute. Session. We shall close the session. If you have any doubt, you can post it in the group. Thank you.
Good morning, students. I hope you are all online and following the classes. Okay. You are almost reaching the end of molecular biology part and its techniques. Okay. So today we are discussing about immunoassays. The two important techniques under immunoassays are radioimmunoassay and ELISA, that is enzyme linked immunosorbent assay. Okay. As you can note the structure of a very famous molecule here. Can you identify what molecule it is? Can you identify? Yes, what molecule has been displayed? I'm just trying to check whether you are all online or just not following the class. Correct. It's an antibody molecule. Correct. So you can see antibody molecule binding to an antigen and all. So all the techniques which we are discussing now are based purely on the immunological reactions. Okay. And here is a picture which represents the technical part of it. How exactly you do it in the laboratory and interpret it. Okay. Okay. So these are the objectives of today's discussion. You will understand the principle of immunoassay, what are the types of immunoassays, what are the principles of radioimmunoassay and enzyme linked immunoassay, various types of ELISAs and steps of ELISA. Basically, you are going into the details of both these techniques. So what exactly is immunoassay? It is an analytical technique. So you are analyzing to find out the concentration or the presence of some biological compound of interest. Okay. So immunoassay is an analytical technique used for the quantification of an analyte. Okay. And the principle used behind this technique is antigen-antibody reaction. Okay. There is high specificity in every antigen-antibody reaction, and this property has been utilized in quantifying the analyte. Okay, and this is known as immunoassay. And so you encourage antigen-antibody reaction here, and in order to quantify the amount of antigen-antibody reaction, you have to monitor it using some labels, okay, or binders. So according to the type of label that you use, the immunoassays have been broadly classified into RIA and ELISA, that is radioimmunoassay if you use the radioactive material as a binder or the label which determines the rate of antigen-antibody reaction, or otherwise if you use enzyme, it is called enzyme linked immunosorbent assay. Okay. So we have used the terms antigen-antibody reaction, right? So let us see what an antigen is. It is a foreign substance which stimulates the production of specific antibody into the body. Okay. It is any foreign substance which is not native to your body. Like you can take the example of a microorganism. Okay. It is totally new to your body. Okay. So when it enters your body, how is it that your body is going to respond to it? Okay. So it is an antigen or it is a stimulus. Okay, which leads to the production of specific antibody or antidote in your body. Okay. So the antidote produced in response to antigen is the antibody. So it is a protective protein produced in response to antigenic stimulus by a foreign substance. Basically, antigen is the stimulus that the body encounters, and antibody is the response which the body produces with respect to that particular antigen. And antibody is usually specific to the antigen which the body encounters. Okay. Okay. The labels used in immunoassay, okay, as you know, it can be the radioactive material or enzyme. Okay. So you can also use chemiluminescent property or fluorescent property of a substance. Okay. So the advantage of immunoassay is that either the antigen or the antibody can be labeled. So you might be thinking what difference it makes whether we use antigen labeling or antibody labeling. Okay, so depending on whether we are labeling antigen or antibody, okay, we have derived varieties of tests under immunoassay, and if at times it may not be possible to estimate the concentration of antigen per se in the body, so at that time you can utilize this opportunity and find out the antibody levels and know how exactly is the patient responding to that particular antigen. So based on the properties or the labels used, the types of immunoassays can be extended to, other than radioimmunoassay and ELISA, you can also extend it to fluorescent immunoassay and chemiluminescent immunoassay, CLIA. Okay. So what are the constituents of a labeled immunoassay? So all these four types of immunoassays which we just mentioned now are labeled immunoassay. They are labeled with some substance or some tracer molecule whom you can monitor. Okay. So what are the constituents of this immunoassay, labeled immunoassay? It should have a specific antibody and it should be labeled. Okay. It should have an antigen which can be a known antigen or unknown antigen. If it is a known antigen, you are trying to find out the antibody levels. Okay. And you have to devise a method to separate the unbound components from immune complex. Basically, you are encouraging antigen-antibody reaction. Okay, there may be too much of antibody or too much of antigen remaining. That is, excess of antibody or antigen has to be removed. Okay, and there has to be a method to remove that. So as a result, we follow too many washing steps in both these procedures. So with every step, there is a washing step in order to eliminate the excess unbound antigen or antibody. And you also have to have a method for detecting the label. Like for example, in fluorescent immunoassay, you have used a fluorescent material. Okay, should have a method to detect that fluorescent signal. Or in case of ELISA, you have attached a enzyme as a label. Okay, so you have to monitor the activity of this enzyme. Okay, and you have to detect the signal generated by this enzyme. How do you do that? You can add a substrate specific to that enzyme, and then you can monitor the amount of product which is produced. That means the amount of enzyme is reflected upon on the amount of product which is formed. Okay, and you need to have a standard for each of these techniques. Okay. So you need to get familiarized with few terms before we move on further. So we keep on using the term solid phase here. Okay, in these techniques, it is usually a micro titer plate. Okay, can you see here? There is a plate with so many wells in it. It usually will have 8 by 12 wells. Okay, so this is referred to as the solid phase. Then what is adsorption? I think you all very well know about the difference between adsorption and absorption. Adsorption is a process of attaching a substance on the surface of some molecule, or it is a process of adding an antigen or antibody so that it attaches to the solid phase or incubation. Okay, so as of now you know that this is the solid phase or the ELISA plate. Okay, so if you are adding some antigen or antibody with an intention that it has to get absorbed to the wells of these microtiter plate, that process is known as adsorption. They are just going and sitting on the surface of these wells. Now what do you mean by washing? This is nothing but simple flooding and emptying of wells with buffered solution in order to separate bound from unbound reagents in ELISA. Okay, I just mentioned you that there are numerous washing steps after every step in ELISA. Okay, or even for that matter radioimmunoassay. This is to ensure that the excess unbound antigen or antibody will be removed and only the bound antibody, that is antigen antibody complex, should only remain, okay, in the plate. If there is any unbound substance, it has to be removed by multiple washing steps. And you know that antigen is the stimulus, antibody is the response produced for that stimulus. Okay. Now, what do you mean by enzyme conjugate? Okay, an enzyme which is attached irreversibly to an antibody is called an enzyme conjugate. Okay, when I mentioned about the labels used in immunoassay, I told you that we can use enzyme, and that immunoassay will be called as enzyme immunoassay or enzyme linked immunosorbent assay. Okay, so the most commonly used enzyme for ELISA technique is horseradish peroxidase. How are you doing? How are you adding this enzyme? You are usually adding this enzyme to the antibody, ensuring that it is irreversibly bound to the antibody. Okay. Now, chromogen. The name itself suggests it has something to do with color, right? So it is a chemical that alters the color as a result of enzyme interacting with the substrate. Okay. When I was discussing about the requirements for the immunoassay, I told you that there should be a method which enables detection of the enzyme activity, right? So what we use here is a chromogen, that is a substance which can change the color at the end of the reaction. Basically, in order to monitor the enzyme activity, you are adding the substrate which is specific to that enzyme and monitoring the production of the product, right? And preferably that product should be a colored one, and this is ensured by the chromogen. Okay. Now, what do you mean by stopping? The process of stopping the action of an enzyme is called stopping. So, you have to at some point stop the action of the enzyme by adding blocking agents. Okay. Then reading. How do you read the enzyme activity? Okay. So, you either use spectrophotometric measurement for the color developed in ELISA. Okay. Tracer is nothing but the labeled compound in an immunoassay. It can be a radioactive tracer in case of radioimmunoassay. It can be an enzyme in ELISA. It can be a fluorescent signal in case of fluorescent immunoassay, or it can be a chemical linked with a chromogen in case of CLIA, that is chemiluminescent immunoassay. Okay, let us learn the process of radioimmunoassay. RIA, okay, radioimmunoassay was developed by Mr. Solomon Benson and Rosalind Yalow, okay, for the estimation of insulin in human serum. This is regarding the history of the radioimmunoassay. Okay, so later it was extended to assay other substances as well. The advantage of this technique is that it can identify very low concentrations of an analyte, okay, to the extent of nanogram or even picogram. So it is highly sensitive, which can pick up the very low concentration of an analyte and is a specific analytical tool. There won't be much interferences, okay, when you use this technique. Okay, these are the scientists who contributed immensely towards the development of this technique, and they also received Nobel Prize for Physiology of Medicine in 1977. What is the principle behind this radioimmunoassay? It is basically a competitive binding assay. Okay, you are radio labeling the antigens, that is antigens are attached irreversibly with a radioactive material. Okay, so you can note that the antigen AG has been added with a star mark. Okay, so this is the radio labeled antigen. It competes with unlabeled antigens, and we assume that these unlabeled antigens are present in patient serum. Okay, so there is no star mark here. So this is the antigen which is present in the patient sample. This is the radio labeled antigen which we are adding externally. Okay, so there is competition between this radio labeled antigen and unlabeled antigen in patient serum for the binding sites on a antibody. Okay, we are adding a limited quantity of antibody, and there is a competition between these two antigens to bind to the binding sites of the antibody. The result being there is a production of antigen antibody complex, okay, with the antigen as the unlabeled antigen. Again you have another set of antigen antibody complex where the antigen is radio labeled. Okay, Then you also have the antigens of the patient serum as well as the radio-labeled antigens separately. Okay. So it is with this radioactive material which is tagged to this antigen, you can trace the amount coming out as a product. Okay. How much of the total radio-labeled antigen has been divided into the bound antigen and the free antigen. Okay. Okay. So the proportion of the antigen-antibody complex, okay, with the labeled antigen and the unlabeled antigen at equilibrium will be equal to the proportion of labeled antigen and unlabeled antigen. Did you understand? So we are basically estimating the concentration of both these complexes, okay, at equilibrium, and projecting the results of the ratio of these two complexes to the original proportion of labeled and unlabeled antigen. Okay. So this labeled antigen is what you have added externally, and unlabeled antigen is the antigen present in serum. Okay. So if you know the concentration of labeled antigen antibody complex, unlabeled antigen antibody complex and labeled antigen, you can find out the concentration of the fourth unknown factor, that is unlabeled, or that is the pure form of antigen present in patient's serum. Okay, it is just a mathematical calculation. If you know three values, the fourth value can be known if you know the relation between those four values. These are the requirements for conducting radioimmunoassay. You need the serum sample, patient sample, okay, serum sample which contains the antigen, okay, and you if we label this as A antigen, you also need antibody which is specifically devised against this antigen. So these are known as anti-A antibodies. You cannot simply call A antigen and A antibody. Okay, when you are labeling antibody against any antigen, you have to start like this, anti-A antibody. Okay. Then you also need radioisotopes for tracing the metabolic reaction, and you require the unlabeled A antigen. Okay. So what you are doing is, initially you are taking the plate, that is the reader plate, and then you are attaching the antibody molecules to the plate. In the next step, you are adding radio labeled antigen in excess. Okay. So what can you expect when you add antigen to already bound antibody? Okay, there can be binding of these radio labeled antigens to specific binding sites on the antibody. Okay, this is what is happening here. But you also can note here that there are some antigens which are unbound. Okay, so in the next step, when you wash the plate, okay, these unbound antigens will get lost. That is, only the bound antigen antibody complex will remain. Okay. Okay. To this bound antigen antibody complex, you add the patient sample, which is supposed to contain antigen which is not labeled. Okay. Initially we added labeled antigen, okay, to make sure that it is bound to the antibody. In the next step, we are adding the patient serum which contains antigen X in question. Okay. So now there happens competition between the radio labeled antigen which was added initially and the antigen in the patient serum which was added in the second step. So the competition between labeled and unlabeled antigens. Okay. So if there is too much of unlabeled antigens in the patient serum, it displaces the labeled antibodies which was attached, labeled antigens which was attached to the antibody. Okay. So we can note here that it is displacing depending on the concentration of the antigen in this serum. Okay. You again wash in the next step to remove any unlabeled antigens. Okay. And then you detect the amount of unlabeled antigen and radio labeled antigen. you will get to know the ratio of labeled and unlabeled antigens bound to the antibody. Okay. So what are the applications? Where do you use this? These are basically used for estimating hormones and proteins which exhibit antigenic properties. Okay. You should know that basically these techniques are based on immunological properties, that is the specificity of antigen antibody reaction. So only if a substance contains these antigenic properties, that is upon its introduction living organisms should be able to produce antibodies to that substance. Okay, so you can estimate such analytes such as hormones and proteins which can exhibit antigenic properties using this technique. And the commonly analyzed particles include the peptides, the structural proteins, hormone receptors, not just the hormones, even the receptors can be analyzed. Okay, the steroid hormones, vitamins, drugs, nucleic acids, and you also can diagnose endocrine disorders based on the analysis for insulin, thyroid hormones, and FSH/LH. And you also can detect cancers by estimating the tumor markers like prostate specific antigen and alpha fetoprotein. You can also do therapeutic monitoring of drugs. Okay, the drugs which are used to treat certain conditions may have narrow therapeutic range within the body. Okay, narrow range within which it is safe inside the body. Okay, if it is too much, it may be deleterious to the body. So that time you can always do therapeutic monitoring of drugs using this technique. Okay, it is very widely used in biomedical research as well. Now, what are the advantages of this enzyme? As I told you, it is highly sensitive technique which can detect even the microgram, nanogram, or even picogram quantities of a substance. And commonly used radio labeled labeling for antigen is iodine 125, which is very widely available, and only microcuries of radioactivity is used. We are not using too much of radio active material. It is only in microcuries. It also has some disadvantages because the equipment and the reagents used in this technique are very costly. You need to buy them. Okay, so only approved laboratories can take up this assay because it is dealing with radioactive labeled antigens and antibodies, basically radio material which is deleterious to the entire living kingdom. Okay, so only approved laboratories which follow biomedical based management appropriately, okay, can take up this assay. So not any lab can do this assay. And the half-life of iodine 125 is only 60 days. So the shelf life is short, and even though only microcuries of radioactivities are used, okay, if it is used too frequently or very widely, the hazards of radioactivity will always remain, and it is a lengthy procedure. It is lengthy and time consuming procedure. Okay, this finishes radioimmunoassay. If you have any doubts, please tell me. If not, I will move on to ELISA. Any doubts? Did you understand radioimmunoassay? If you don't have any doubts, I have few questions. Please name 2 applications of radioimmunoassay. Applications of radioimmunoassay. Where do you use this technique? 2 applications of radioimmunoassay. Yes, come on. No applications? Estimation of hormones and proteins. Okay. Very good. That exhibit antigenic properties. Very good. Estimation of hormones. Right. Only hormones. I told you about therapeutic drug monitoring, tumor markers for cancer. Right? Okay. Let us move on with rather more important technique, that is ELISA. The expansion of this term ELISA is enzyme linked immunosorbent assay. Enzyme linked because we are using enzyme as the label here. It is linked, okay, immuno because it is based on the immunological properties. Sorbent, that is adsorption, it is based on the adsorption of the antigen or the antibody onto the ELISA plate. Assay, we are assaying the entire principle or the procedure. So it is known as enzyme linked immunosorbent assay. This is the ELISA plate, and this is how we can notice the color changes following the set of procedures to be followed. Okay, you can note that you can see so many shades of blue here and so many shades and some yellow colored wells as well. Okay, one indicates positivity, the other indicates negativity. These are samples from different patients. Okay, the samples from different patients can be pooled and can be assayed at once. Okay, you can note that few of the results are positive, few of the results are negative. Okay, and even in positivity you can note the shades of blue, that is depending on the concentration of the antigen or the antibody. If it is lighter, probably lesser concentration. If it is darker, probably higher concentration. ELISA is a quantitative immunological procedure in which the antigen-antibody reaction is monitored by measuring the enzyme activities, and as a result their concentrations can be measured. Okay, it is very similar to radioimmunoassay, quantitative because you can estimate the quantity of the substance you are analyzing. It is an immunological procedure because it is again based on antigen-antibody reaction specificity. Okay, so what are you doing? You are attaching the enzyme to the antigen or the antibody, and then you are assaying the enzyme activity and thereby measuring the concentration of the antigen or the antibody in the patient sample. It is a plate based assay. I just had showed you about the ELISA plate. So we do the assay in a plate, okay, known as ELISA plate. So it is a plate based assay designed for detecting and quantifying. It is qualitative as well as quantitative. You can either just detect that there is this type of antibody in this sample, or you can also quantify, yes, there is this antibody and this much of antibody is present in patient sample. Okay, so it can be both qualitative and quantitative. Okay, for estimating peptides, proteins, hormones, tumor markers, antibodies, growth factors, etc. If you notice any laboratory results, okay, so probably you would have asked for antibody, like for example, antibodies against viral antigens, okay, will be expressed as either titers or just present or absent. So it can be a quantitative result or qualitative results. And for example, tumor markers, okay. Suppose you are taking a patient with a neoplasm to get their tumor markers assay. Okay, the results will be displayed as, like for example, prostate specific antigen is present as dash concentration. Okay, and you can always assay hormones. Okay, using this technique. It is a simplified and modified version of radioimmunoassay. While we were discussing the disadvantages of radioimmunoassay, we discussed that it is a little complicated and lengthy procedure. Okay, So this procedure was simplified by modifying a few of its contents, okay, and the result is ELISA. The enzyme is used as a label in ELISA in place of radioactive material which is used in radioimmunoassay. Okay, that is the main difference. And ELISA is more sensitive than RIA, as it is radioimmunoassay itself was very sensitive, which could detect to the level of picograms of an antigen or antibody. Okay, this ELISA is even more sensitive than radioimmunoassay, and the added advantage of no risk of radiation hazard. Okay, because we are not using any radioactive material here. The tracer which we use here is an enzyme. Okay, and it is a naturally occurring substance. Okay, what is the principle of ELISA? Again, it is based on immunochemical properties of the antigen antibody reaction. Particular antigen can combine with only specific antibody. Okay, so it can be compared to the lock and key model of enzyme action, where the antibody acts as a lock and antigen acts as the key. Okay, so the key fits into the lock specifically. Okay, so how do we do that? The binding reagent, okay, either the antigen or the antibody which is irreversibly bound with the enzyme is initially immobilized onto a stationary solid phase. Okay, and this is known as ELISA plate. Okay, on the ELISA plate you are immobilizing the antigen or the antibody. Okay, and this ELISA plate is known as the stationary solid phase. In the next step, it is then complexed with an antibody linked with the enzyme. Okay, so you are first immobilizing the binding reagent onto the ELISA plate. In the next step, you are adding an antibody which is attached with an enzyme. Okay, what do? The detection is accomplished by assessing the activity of the conjugated enzyme. Okay, so how do you know the activity of the enzyme? By adding a substrate which can be converted to a colored product. Okay, so the most crucial element in the entire detection strategy is the highly specific antigen antibody reaction. Okay, if suppose you dissect out one well in the ELISA plate like this, okay, it looks like this, something like this. Okay, so this well is added with the antigen. The antigen is immobilized onto the well. Okay, so it should not move, it should just get stuck to this well. The next step, you are adding a antibody which is specific to this antigen. Okay, usually this antibody carries the enzyme. Okay, in certain modified procedures of ELISA, we can use two different antibodies, that is the primary antibody which is against the antigen. Okay, if it is directly attached with the enzyme, it is known as direct ELISA. If you also have a secondary antibody which is directed against the primary antibody, okay, and it is this secondary antibody which is attached with the enzyme, okay, it is known as indirect ELISA. So the only difference between direct and indirect ELISA is, in direct ELISA you have the antigen and then specific antibody directly linked with the enzyme. Okay, in case of indirect ELISA, it is antigen which is immobilized onto the well of the plate, then you add the antibody. Okay, in the next step you add a secondary antibody which is directed against the primary antibody, and it is this secondary antibody which is, okay, linked with the enzyme. So how do you monitor the activity of this enzyme? Like for example, here we have used horseradish peroxidase. So you have to add a substrate which can be acted upon by this enzyme. Okay, and as a result it can produce a colored product. Okay, so you are assaying the quantity of the color produced. Okay, so and this would depend upon the concentration of the enzyme. This concentration of enzyme will depend upon the amount of secondary antibody binding the primary antibody, and this depends upon the amount of antigen which is present in the patient sample which was added to the well and immobilized. I hope you understand. I will explain you again. Okay. For example, the sample which was immobilized on the ELISA plate contains different types of antigen. Okay. We are not interested in all these antigens, but one antigen. Yes. Okay. If you are trying to estimate the quantity of one specific antigen, okay, you have to immobilize the sample onto the well. Then you are adding the primary antibody against this antigen. Okay. Please note here that this antibody is not binding to any other antigens. It is highly specific to the antigen of interest, the primary antibody. Okay. It can be directly linked with the enzyme and you can monitor the enzyme activity, or if the concentration of the antigen here is very less, okay, we can amplify the final signal produced by adding a secondary antibody directed against this primary antibody. Now this secondary antibody is labeled with enzyme, like for example horseradish peroxidase. Now you have to monitor the activity of this enzyme by adding a substrate, okay, which will go and bind to this enzyme and then it will form a colored product. You can assay the amount of color present in this colored product, like how you used to do colorimetric assays, okay, and this correlates with the concentration of the antigen directly. So your entire intention is to know the amount of antigen present in this sample, a particular type of antigen, not the other ones. Okay. So you are using too many procedures in order to produce a color, and this final color is proportional to the concentration of that particular antigen in that sample. So in order to do all this, we need equipments, ELISA equipments known as ELISA reader and ELISA washer. Okay, we need the ELISA plates on which you are immobilizing the antigen or the antibody, and you also require certain reagents which contain the antibodies, okay, and the secondary antibodies which are labeled with enzyme. This is the ELISA reader on which we perform the assay. Inside the ELISA reader we have either spectrophotometric unit which can measure the final color. Okay. Can you notice here that there is an ELISA plate here? Okay. We usually use 8 by 12, 8 into 12 well-sized plates. Okay. So once you perform all the antigen antibody reactions, then the final color produced will be fed into this equipment, and it is this equipment which will read the optics or the concentration, the intensity of the color, and thus the concentration of the antigen. So what reagents do we use in ELISA? We use buffers, 3 different types of buffers. One is known as the coating buffer, the other is known as the washing buffer, and the final one is the blocking buffer. So coating buffer is used to coat the surface of the ELISA plate. Washing buffer, okay, washing has to happen after every step in ELISA, and we are not just washing it with water, okay, we are taking a buffered solution. And then blocking buffer, this is to stop the enzyme activity at after a particular time, okay. Then you also need a enzyme, like for example horseradish peroxidase. You need a chromogenic substrate, that is a substrate which can produce color once getting converted to product, and then we require a stop solution. Okay, so this is how the ELISA plate looks like. It is usually flat bottom. Okay, these circles are the wells, okay, which are 3-dimensional structures. Okay, these are usually 96 well plates because it will have 12 wells in one row, and it will have 8 such columns. Okay, so 12 by 8 is 96. 96 well plates. Okay, and each of the well will hold 350 microliter of sample. Note that it is not ml, it is microliter. So these wells are really tiny. These are made up of polystyrene or polyvinyl chloride, okay, and it is coated with inactivated antigen or antibody. Please note that we can assay either antigen or antibody present in the patient sample, okay. Now the coating acts as a binding site for the sample antigen or antibody, okay. So the plates which are pre-coated with antigen or antibody are also commercially available. Suppose you are dealing with assaying of one particular antigen or one particular antibody, specifically you can buy those plates which are coated with those antigens or antibody and use it for repeated assays. It maintains consistency, it minimizes the edge effects and provides optimal conditions. Okay, why we using polystyrene or polyvinyl chloride is because it is really inert, okay, and it provides optimal optical conditions, okay, because finally it has to be read out by a spectrophotometric system in order to tell us the concentration of the analyte present. Now we also require the multipipette. You have used automated pipettes in your biochemical analysis, right? So we use multi-pipettes here, wherein with single plunging action you can take out the sample in 8 different tips, okay. Can you notice here that there are 8 different tips, since the most commonly used wells contain 8 by 12 wells, okay. So once you take out the reagent or the sample, okay, you can add all these reagents at a time to all the 8 wells, okay. So this is another equipment required for ELISA, that is microplate washer, okay. So for washing the plates to remove the excess unbound antigen or antibody, after every step we use this washer. It is very efficient and it is usually very low carryover, that is if it doesn't wash it properly, the unbound antigen which is still remaining attached to the ELISA plates will be carried over for the next step, okay. But this is eliminated in the using this washer because it is highly efficient in removing all the unbound antigen or antibody. Washing can also be done using manual methods, but this is more accurate than the manual washing device. Then you also use ELISA buffers. What is a buffer? Can you just tell me what is a buffer? What is a buffer? Maintains pH, okay. Rather you can say buffer that it resists the change in pH to a certain extent, okay. So when you are coating the ELISA plate, when you are blocking the ELISA plate, when you are washing, okay, you are not using the simple distilled water, rather you are using buffers which are chemically similar to the reagents that you are using, okay. So you use several different buffers during ELISA. One is used for coating the ELISA plates initially, another for blocking, okay. Why this blocking? Okay, because you don't want the other wells to get contaminated, okay. And then another for washing and perhaps another for sample and antibody dilution. So even for dilution we are using buffer. These buffers can be produced in-house within the laboratory, okay, or it can be commercially obtained from the suppliers, okay. So what are these coating buffers? Coating is a process where suitably diluted antigen or antibody is incubated, okay, until it is adsorbed onto the surface of the well. You very well know that there are multiple wells in an ELISA plate. Okay, you want to coat this ELISA plate with a buffer, okay, which contains the antigen or the antibody, and you make sure that this antigen or the antibody gets adsorbed or gets attached to the surface of the well. This adsorption occurs passively, okay, as a result of hydrophobic interactions between the amino acid side chains and the plastic surface. Okay. So what is the role of this coating buffer? It stabilizes the antigen or the antibody coating. Okay, it makes sure that it sits there, it doesn't move anywhere. Okay, it maximizes the adsorption, and there is optimization of interaction with the detecting antibody. The most common coating buffers used are bicarbonate buffer at a pH of 9.6. Now, what are these blocking buffers? These buffers prevent non-specific binding of detection antibodies. Okay, and there are two types of blocking buffers. We can use either proteins, which can act as permanent blocking agents, or we can also use detergents which can block temporarily. Then the third type of buffer which we use is washing or the diluting buffer. Okay, we usually use PBS with between 20. It is to remove the unbound materials. In whatever reagent you are adding or the sample you are adding, it will contain excess of antigens or antibody which is totally unrelated to the assay. Okay, so these if it is unrelated to the assay, it will not bind to the antigen or the antibody which is coated onto the surface of the well. They remain unbound or free. Okay, so we have to remove this free materials, okay, using this washing buffer. This washing is repeated 3 to 5 times between each step in ELISA, not just single time. It is 3 to 5 times. Okay, the excess wash solution must be removed in the final wash step. Okay, since you are using multiple washing steps, okay, before you submit the final product into the ELISA reader, you have to remove the washing buffer excess which is present, okay, in order to prevent the dilution of reagents. Now, we are using enzymes as a label, right? So, what are these? These are naturally occurring ones which catalyze biochemical reactions. And how do you select the enzyme? What type of enzyme is required for that particular assay will depend upon the substrate molecules, the ease and speed of detection, the stability of the enzyme reagent, availability. Do we have to go in search of this enzyme, or is it easily available? Okay, and it also depends on the cost of the enzyme. So, considering these factors, the enzyme labels which are commonly used are horseradish peroxidase, alkaline phosphatase, glucose 6-phosphate dehydrogenase, beta galactosidase, acetylcholinesterase and catalase. The first two, that is horseradish peroxidase and the alkaline phosphatase, are very popularly used in ELISA assays because they have high turnover rates, high sensitivity, that is they can detect the very minute concentration of an antigen or antibody, and there is it's a easy process to detect these enzymes because the substrates required for these enzymes are easily and plentifully available. Now, what substrate you use? Okay, so the selection of substrate depends upon the enzyme that you use. Okay, if you are using horseradish peroxidase as the enzyme, okay, you use ABTS or OPD or TMB, that is commonly used one is tetramethylbenzene as the enzyme. If you are using alkaline phosphatase as the enzyme, then the substrate used is paranitrophenyl phosphate, the disodium salt. Again, the choice of the substrate, see, for horseradish peroxidase you have 3 different substrates here. Okay, so which one will you choose will depend upon the requires assay sensitivity and the instrumentation which is available for signal detection. Okay, whether you have the fluorometric assay or spectrophotometric assay. When I showed you the ELISA reader, I showed you that there is a optical system inside which enables it to detect spectrophotometrically or fluorimetrically. Okay, so depending upon what optics you have, okay, you also have to select the substrate. Now, ELISA antibodies. The antibodies which are used in ELISA can be monoclonal antibodies or polyclonal, or it can be a combination of these two. What are monoclonal antibodies? These are specific against the single epitope of an antigen. Okay, if you have listened to the immunology class properly, you will know the meaning of this epitope. What is an epitope? Can someone tell me what is an epitope? Epitope of an antigen. What do you mean by epitope? Okay, it is the antigen determining site on the antigen. Okay, so the entire antigen may not be immunoreactive, or it is the antigen detecting part of the molecule which is called the single epitope. It has the capacity to raise the production of antibody. Okay, so if a particular antibody molecule is acting against a single antigenic determinant, it is called monoclonal antibodies. Okay, polyclonal antibodies are those which can act on various antigenic determinants of an antigen. Okay, these polyclonal antibodies can be used as capture and detection antibodies, which I will be telling you later. Now, how do you describe the interaction between the antibodies and their antigens? It is highly specific. Okay, that particular antigen combines with the specific antibody only. There is too much of affinity between the antigen and the antibody, and there is avidity. Okay, so how do you do this ELISA? Okay, so once you know the requirements for ELISA, we should know how to perform the procedure. Okay, so first we have to immobilize the antibody onto a solid surface. Okay, can you notice a ELISA plate well here? Okay, this is the solid surface onto which you are immobilizing the antibody. Can you see these Y-shaped structures? These are the antibodies, the first antibodies which you are immobilizing on the solid surface. Then the next step, you are adding the sample which contains, you are adding the patient sample which contains the protein of interest, which you are interested in detecting this protein. Okay. The next step, there is complexation of antigen with the primary antibody. Okay. So we had added the antigen. Now these antigen will go and bind on the antigen binding sites of the antibody. So can you notice here that there is antigen antibody complex. Few of the antibodies may remain vacant if there is lesser concentration of antigen. Okay. The next step, you add enzyme linked secondary antibody. Okay, the pink colored structures here are the secondary antibodies linked with the enzyme. Okay, then reaction of in this step, so when you have added the secondary antibody being linked with the enzyme, it will go and bind to that particular antigen. Okay, so at this point of time you have a solid surface with immobilized antibody molecules. Okay, and then you have added the antigen molecules which are bound to this primary antibody. Again, subsequently you have added the secondary antibody linked with the enzyme. So wherever you have antigen, you also will have a secondary antibody with enzyme. See here, there is no antigen, so you cannot find the secondary antibody also. Okay. Then in the next step, the reaction of substrate with this conjugated enzyme is assayed in order to produce the colored product. Okay, so you have the enzyme attached to the secondary antibody now. Now you have to in order to measure the concentration of antigen, you need to measure the activity of this enzyme. How do you do this? You add a substrate and then quantify the amount of product produced. Usually this product is colored and it can be assayed in spectrophotometer or fluorometer. Okay, so to summarize, you are coating the well with antibody known as primary antibody. You are incubating it for a definite period of time so that there is bonding between this antigen and antibody. Okay, there are few antibody molecules which are not bound. Okay, so when you wash the ELISA plate at this point, you can remove the unbound antibody molecules. The next step, you are adding the secondary antibody, okay, which is also directed against this antigen. Okay, so this antigen will have multiple antigenic determinant sites. Okay, so one of the site is recognized by this primary antibody, other antigenic determinant is recognized by the secondary antibody. So the site which it recognizes is the binding site. The secondary antibody is linked with an enzyme. Okay, so in the next step you again wash to remove any unbound secondary antibodies, and then you add the substrate specific to this enzyme, bound enzyme, and then there will be color change, and finally you will measure the intensity of the color of the samples in each of these wells at 450 nanometer. Okay, so we kept on telling you the patient sample. What all samples can you use? You can either use serum or the CSF, sputum sample, urine sample, semen sample, or the supernatant of culture, or even stool sample. Okay, so you can assay the amount of antigen or antibody in any of these samples using this ELISA technique. Okay, these are the various types of detection. This is a colorimetric type of detection, okay, where the substrate will produce the colored product, okay, and this colored product will be in the visible range of the electromagnetic spectrum, that is within 400 nanometer to 700 nanometer. Only then you can say it using the colorimeter. Okay, or it can be chemiluminescent immunoassay, where it produces the light and it can be assayed using chemiluminescence technology, or you can also use the fluorescent material, okay, which will produce light and assayed using the principle of fluorescence. So what are the types of ELISA? So based on the procedure that we use, the procedure followed, whether we following 1 step, 2 steps or 3 steps, you have various types of ELISAs. It can be non-competitive ELISA, competitive ELISA, or multiple and portable ELISA. Under non-competitive type, you also have direct, indirect and sandwich ELISA. Okay, so it is these three which are used very widely: direct ELISA, indirect ELISA and sandwich ELISA. What is direct ELISA? Here an antigen is mobilized onto the well of an ELISA plate. You have a ELISA plate and you have immobilized an antigen, okay, and you have allowed sufficient incubation time for it to get absorbed onto the surface of the plate. Okay, so the antigen is then detected by an antibody which is directly conjugated to the enzyme. Okay, you can note here that the plate is coated with the antigen and you are adding an antibody which is linked with an enzyme. There is no secondary antibody involved in direct ELISA. So you have only one antigen, one antibody and one enzyme in direct ELISA, and you assay the concentration or the activity of this enzyme using a substrate which produces a colored product. This is direct ELISA, which is simple. The advantage is that it is quick and less prone to error, okay, but the disadvantage is that the antigen immobilization is not specific, it is less flexible, no signal amplification, okay. If you are suspecting that the antigen or the antibody concentration in the patient sample is very less, you cannot use direct ELISA because there is no signal amplification, because we are using only one antibody here. Now, what do you mean by indirect ELISA? The antigen will be absorbed at the surface of the ELISA plate. Okay, the detection happens in two steps. That is, the primary antibody is added, okay, which binds with the antigen. Again, in the next step, we are adding a secondary antibody directed against this antibody, okay, and it is this secondary antibody which is linked with the enzyme. Okay, again you measure the activity of this enzyme. This is known as indirect ELISA. So in direct ELISA, we had only the primary antibody. In indirect ELISA, you have primary and secondary antibody. That is the only difference. Okay. Why are we taking two sets of antibodies? In order to enhance the intensity of the signal. Okay. Indirect ELISA is more economical, high sensitivity, it is very flexible, versatile, okay, and the signal amplification is really huge. Okay. The disadvantages being there can be some background noise, it is a longer procedure, okay. There is additional incubation step for the secondary antibody. Okay, this is how you can represent the procedure followed in indirect ELISA. So, do you have any doubts between direct and indirect ELISA? Please ask me. We are now moving on to the sandwich ELISA. If no doubts, I'll just carry on. So this is direct ELISA, where the antigen is bound to an antibody labeled with an enzyme. This is indirect ELISA, where you have two different antibodies, okay, and then the enzyme tagging is happening. Sandwich ELISA is used for antigen detection, okay. The first two types, that is direct and indirect ELISAs, were basically used for antibody detection. Here you are detecting the antigen, okay, which requires matched antibody pairs, okay, known as the capture and detection antibodies. Let us see what is this. The first antibody, called the capture antibody, is coated onto the plates of the ELISA well, okay. So it binds with the antigen in the sample. So once you add the capture antibody to the plate, then you add the sample, which is supposed to contain the antigen of interest. Like the antigen can be a tumor marker, hormone, serum protein, etc. Okay. So it is immobilized and then it is detected using direct or indirect ELISA. Okay. This entire process is known as sandwich ELISA. So you have the capture antibodies which will capture the antigens, okay, and you have the detecting antibodies, primary and secondary antibodies. So the second antibody, which is known as the detection antibody, okay, it creates a sandwich with the primary antibody added, the antigen, and then the radio labeled or the enzyme labeled antibody. So the antigen here is sandwiched between two antibodies, correct? The antigen has been sandwiched between capture and detection antibodies, hence the name, okay. So this can be used only when the antibodies can recognize different epitopes of the same antigen. I am telling you repeatedly, epitope is nothing but the antigenic determinant component of an antigen, okay. So there can be various epitopes in a single antigen, okay. So the antibody used in sandwich ELISA should be able to detect various epitopes of an antigen. That is a prerequisite, okay. It is very sensitive and highly specific compared to other types of ELISA. It is flexible and it is the best for analysis of complex samples. Okay. So this is how you carry out the sandwich ELISA using capture antibody and then adding the antigen, and then washing to remove unbound materials, and then you are adding the antibody which is labeled with the enzyme and detecting. Okay. We will continue with the competitive ELISA in the next class. Please read up. We will discuss in the next class.
students. So we will continue with yet another topic related to the abdominal cavity. Today's topic will be related to the one of the venous channel, one of the important venous channel in the abdominal cavity, that is the portal vein. Now, just like any other topic related to anatomy, so it is very much relevant to be aware of what type of kind of question can you or you can expect in examination. So keeping that in mind, related to portal vein, so this can be, you can expect this one as a 10-marker question, as well as can also be asked as a 5-marker and even as a 3-marker questions also. So this is how you can expect a question related to portal vein, as to you have to describe the portal vein, how exactly it is formed, what would be the course of the vein, since it is a venous channel. So what would be the course of the vein, then what are its tributaries, draining channels into the portal vein, and even one of the most important aspects of the portal vein would be the anastomosis. This is called as a portal cable or portosystemic anastomosis. This itself can be a separate 5-marker or 3-marker question. And last but not the least would be the upper aspect related to this. So this is how you can expect a question in examination. Now, we will look into slighter details about this particular venous channel. This portal vein, since it forms a very important venous channel, as I told you earlier, why exactly is it an important venous channel in abdominal cavity? Now it has its own reasons. The reasons can be about 3 to 4 reasons. It can be put forth as 3 to 4 important reasons why it forms a very important venous channel. As you can make out in the diagram, you can still make out the impression of the duodenum, and with its concavity you can see even the pancreas as well. Of course, the neck and the part of the body of the pancreas has been reflected so that we can get a better visualization of the vein over here. So this vein here, this channel, this is called the portal vein. So behind that you can make out of the background, the larger vein is the inferior vena cava, but or you can say the channel between the duodenum and the inferior vena cava. So this is called the portal vein. Now it is approximately dimension when it measures almost about 7.5 centimeter long. So it almost approximately 7.5 centimeter long. Now, as I said, important venous channel because of these reasons. One, this venous channel, portal vein, it will drain the venous blood from the abdominal as well as the pelvic parts of the alimentary tract or the GI tract. So the GI tract related to abdominal cavity as well as the pelvic cavity. So almost most of the gastrointestinal tract is drained by the portal vein. So that's why forms one important reason for to be called as an important venous channel. Apart from that, it also drains gallbladder. Also, it will drain even the spleen, which is seen over here, and as well as the pancreas. So all these 4 draining areas, so almost covers most of the gastrointestinal tract along with its organs in and around the abdominal cavity. So that's why it forms an important venous channel. So that is how we can sum up the important feature or why it forms an important venous channel, because it drains the GI tract, most of the majority of the GI tract, abdominal and pelvic cavity. It will drain the spleen, it will drain the pancreas, including the gallbladder. And ultimately, whatever the venous blood, this venous channel portal vein drains, it will convey it ultimately to the liver. So that is how the end of the portal vein functional aspect comes to an end. So it will convey all the blood from draining areas to the liver. So that is how we can that is as an introductory mode to this important venous channel, that is the portal vein. Now we will see some important but more relevant features again with respect to portal vein as well. Apart from being called as an important venous channel because of its drainage areas, extensive drainage areas of the abdominal cavity, including those features, there are again certain important features which can be related to the portal vein. Like for say, 80% of the blood that flows to the liver is conveyed by the portal vein itself. So remember that 80% of the blood which is in the liver, the liver gets 80% of the blood from the portal vein itself. Whereas the remaining 20% of the blood, the liver receives, it will receive in the form of hepatic artery. From hepatic artery, rest of the 20% of the blood that liver gets is from hepatic artery, whereas 80% of the blood is conveyed to the liver by the portal vein itself. So that forms a very important aspect of the portal vein. Whereas other than that, it is said that almost if you compare it with the total volume of the blood, we approximately say for say 5 liters of the blood of total volume, from 5 liters of the blood, almost 1/3 of the total volume of the blood is found to be present in the tributaries of the portal vein itself. So that is how it forms again one more important aspect of the portal vein. So almost like if you compare the 5 liters of volume wise, so almost more than 1.5 liters of the blood will be present in the tributaries of the portal vein itself. So that is how extensive amount of volume of the blood to be seen in the portal vein. And as well as one other feature would be that portal vein, unlike any other veins, it is said to be devoid of the walls inside that. So that is how this image again shows the entire GI tract almost, most of the GI tract, including the small intestine of course. So you can make out over here, the portal vein goes, this channel is the portal vein. We will see the formation and all those things in the next slide. So this is not the portal vein conveys. So conveys all the venous blood from the GI tract, most of the GI tract. Whereas the other feature which can be related important feature, categorized as an important feature related to the portal vein would be like portal vein will carry or convey the products of digestion in the form of carbohydrates or the protein or other nutrients from the intestine, small and large intestine, small more particularly. So these metabolic products will be conveyed by the portal vein to the liver for the metabolic process to happen. So that will be conveyed by the portal vein. Even that includes even the RBCs, further again for the further the overall RBCs, again that also from spleen will be conveyed to the liver, as from spleen will be conveyed to the liver by the portal vein itself. So that is again one of the important feature. Other would be one more important relevant feature would be just like since this portal vein will be running parallel with the hepatic artery, so it will follow the branching pattern. If you look at when we look into the branching pattern of portal vein, so then you can imagine the next slide. So it divides has the same branching pattern just like the hepatic artery, and whatever the blood that which is carried by the portal vein from the GI tract or the draining areas, it will be conveyed to the liver into the sinusoids of the liver. Whereas from the liver, the hepatic vein take the blood to the inferior vena cava. So that is how we can say ultimately the beginning and the ending of the portal vein happens. So it will drain all the venous blood from the draining areas. It will convey to the liver in the form of branching pattern, exactly more or less similar to the hepatic artery. So it will be conveyed to the sinusoids, and from there it will be drained by the hepatic veins into the inferior vena cava. So that is how the channel conveys the blood. Whereas one more important aspect that is worth remembering would be, it is said that this portal vein portal vein begins as a capillary bed in the gut. We know the draining area. So since we know the draining area of the portal vein, that is an exclusively GI tract. So there in the GI tract it begins as a capillary bed in the gut, that is GI tract, and as it terminates like an artery in the hepatic sinusoids. So it's worth remembering the statement related to the portal vein. So it begins as a capillary bed in the gut, whereas it ends as a sinusoids in the liver. So that is again one of the important statements worth remembering related to the portal vein. After these introductory and then certain important features related to the portal vein, now we will see how exactly the portal vein is formed and what would be its course of the portal vein and what happens to its further if you trace it as a branching pattern of the portal vein. Now, first and foremost would be the formation of the portal vein. Now, portal vein is said to be formed by union of, as you can make out from the diagram, so it is formed by union of the splenic vein as well as the superior mesenteric vein. So superior mesenteric vein and the splenic vein, they join to form or give rise to the portal vein. So that is how the formation of the portal vein can be described. But if you wish to relate it to the bony structure, so it is said to be like related this fusion or union happens, or this formation of the portal vein happens at the level of the second lumbar vertebra. So that is where the exact location of the portal vein can be mentioned or related with respect to the vertebra, the second lumbar vertebra. So that is how it forms. Now once it is formed, so as you can make out in the diagram, so once it is formed, it will ascend upwards. So it will ascend upwards, particularly as you can make out in this diagram, just a slightly magnified image. Once after its formation by union of splenic vein, this is the splenic vein. So as I told you earlier, so you can make out or still make out the image of the impression of the duodenum can be made out. You can make out within the concavity of the duodenum, pancreas is also seen. But only thing is that the part of the pancreas with the neck and the body has been reflected, the second reflected, so that you can see the better view of the portal vein over here. Now, so once after formation of portal vein, the union of superior mesenteric vein and the splenic vein over here, so behind the neck of the pancreas, so it will ascend upwards in such a way that it will be initially behind the pancreas, then further it will run vertically above, being or staying behind the duodenum, especially the first part of the duodenum, and further it ascends upwards to be seen. So if you further trace this portal vein slightly above, we see the portal vein, course of the portal vein to be related to the lesser momentum in relation to the stomach. So there we can make out it. That is where you can trace it. Now in this image again, so after the formation, so you can still make out the duodenum here. So the lower part of the image is not visible, but once the portal vein crosses the duodenum, first part of the duodenum, we will make out, or we can make out the portal vein within the right margin of the lesser momentum. What we can make out is this stomach to be the lesser curvature here. Now it is attached to the lesser curvature. This structure represents the lesser momentum. So this being the right margin of the lesser momentum. So where we can make out the portal vein. So that is where the portal vein courses through after its formation, and further it will end. So where it will end? So if you further trace it, almost you can. This part of this is the under surface of the liver, what you can make out, which has been slightly lifted up, so that we can get a better visualization. So the liver, under surface of the liver has been reflected upwards, so that we can see the interior surface of the liver. So here you can make out the portal vein again. So portal vein, so seen earlier in the lesser momentum right margin, and then ultimately ends in the porta hepatis. So by this time you might be aware of what exactly the porta hepatis. So that is the higher region in the liver where instructors enter and come out of the liver. So in this region is called the porta hepatis, where you can make out the portal vein to be terminating into right and left branches. So it ascend upwards. So initially it will be seen to be present behind the neck of the pancreas, because the part of the neck of the pancreas has been dissected out. So it will be initially behind the neck of the pancreas. Further it will ascend upwards to stay behind, or it will run behind the duodenum, specifically the first part of the duodenum, and further ascends upwards to be seen in the lesser momentum, right margin of the lesser momentum. So this image represents, as I told you earlier, the first part of the duodenum, which is second, third, and the fourth part. So it will be seen as a when it ascends upwards behind the duodenum, further ascends upwards to be seen in the right margin of the lesser momentum over here. Further, it will terminate or will end the course, it will end its course after reaching the porta-hepatic region of the liver, on the under surface of the liver, where it will end by terminating into right and left branches. So that is how the formation and course of the portal vein can be described. Now this is again where it ends up. Now as you can make out here, porta-hepatic, just for a better understanding. So this represents the bile duct, then the artery, hepatic artery, and the portal vein. So this is how it ends up by dividing into the left and the right branches of the portal vein. So that is how we can mention as a termination of the portal vein. Now briefly about the branches, what happens to the branches, and how exactly specifically they end up into the liver. Now if you take up the first branch, the terminal branch of the porta-hepatis, the portal vein, so you can make out here as dividing into right branch and the left branch of the portal vein. Now even though it is a vein, we describe it as a branch since it is in close approximation to the hepatic artery, or it follows the branching pattern of the hepatic artery. So even instead of tributary, we mention this one as a terminal branches of the portal vein itself. Now dividing into right branch and the left branch of the portal vein. Now if you compare, in comparison, the right branch of the portal vein is said to be wider and shorter. So whereas the left branch is considered to be longer and narrower. So that is how the difference between the right and the left branch of the portal vein can be mentioned. So right will be shorter and broader. It will receive. Now this right branch will ultimately end into the right lobe of the liver. Before ending the right lobe of the liver, it will even receive the cystic vein, that is the vein draining from the gallbladder, or cystic vein. So that will drain into the right branch of the portal vein. So mind you that. So right branch of the portal vein, it will receive the cystic vein as well, and then it will drain to the right lobe of the liver. Whereas what happens to the left branch of the portal vein? Now since it is quite longer and narrower, now this left branch, no doubt the left branch of the portal vein has to enter into the left lobe of the liver. Before entering into the left lobe of the liver, it will receive no doubt even give branches through the caudate and the caudate lobe of the liver as well. Now the more important would be, so it will, what happens is, if you look into this diagram, so portal vein, this is the right branch of the portal vein, the left branch. So now as you can make out, right branch is visible in the cystic vein and ultimately leading to the right lobe of the liver. Whereas we are tracing the left branch now. To trace the left branch of the portal vein, as you can make out, this left branch ultimately goes and fuses with the two other ligaments. That is, one is called the ligamentum venosum, as it is marked as a broken line over here, ligamentum venosum, and there is one more broken line which represents the ligamentum teres. So it will almost fuse with that. So left branch basically unites with the ligamentum teres and ligamentum venosum. Now what would be important is the ligamentum teres separatus. So this ligament, along with this ligamentum teres, running with that will be a certain group of veins which are called as para-umbilical vein, which drain the umbilical region. So para-umbilical vein. So they also run with the ligamentum teres separatus. So these are para-umbilical veins. They drain into the left branch of the portal vein before ending into the left lobe of the liver. So that is how the left branch of the portal vein ends into the left lobe of the liver. So just like the right branch which ends into the right lobe of the liver, before ending into right lobe, it will receive the cystic vein, whereas before the left branch of the portal vein, it will receive the para-umbilical vein and will end up into the left lobe of the liver. So that is on the termination of these two specific branches of the portal vein. Now just for your better understanding, this is the undersurface of the liver wherein you can make out this is the left half, the right half of the right lobe of the liver. You can see the gallbladder as well. Porta-hepatic region over here. Now this is the fissure, which is called the fissure for ligamentum teres hepatis, and this fissure more towards the posterior aspect would be the ligamentum fissure for ligamentum venosum. So that is where the portal vein comes in contact with these two ligaments, whereas running with the ligamentum teres is through the para-umbilical vein that they relate to the left branch of the portal vein. So that is how in relation to these two ligaments. So that is how the formation, the course, and you can say the branching pattern or the termination of the portal vein. So same is slightly magnified, so that we can better understand the ligamentum venosum, the ligamentum teres hepatis, and into the teres hepatis is the para-umbilical veins. So that is now in right branch, cystic vein leading to the right branch. So that is what happens with respect to the, I can say, before the portal vein enters into the liver, so that will be the course of the portal vein. Now, what happens to the portal vein once it enters or once it goes into the structure or into the parenchyma of the liver? So that is called as an intrahepatic course of the portal vein. Now, so as it is very much evident from the diagram. So once the portal vein has a right branch and the left branch of the portal vein, when they enter into the substance of the liver or the parenchyma of the liver, it will divide and redivide to form smaller and smaller veins and venules. Now ultimately they end up into the spaces between the hepatocytes or the liver cells or the hepatocytes. So the spaces which are present between the hepatocytes or the liver cells, they are called as hepatic sinusoids. So that is where the portal vein ends or pours or conveys the blood which it has drained from the GI tract into the hepatic sinusoids. So once it enters, the portal vein divides and redivides, becomes smaller and smaller until it ends up into the hepatic sinusoids. Now what happens in the hepatic sinusoids? As you can make out from the diagram, so this represents the liver or the hepatic sinusoids. Though the hepatocytes has not been shown, so only thing is that has been shown over here is the hepatic sinusoids or the liver sinusoids, as I mentioned in the here, as the liver sinusoids or the hepatic sinusoids. Now what happens after that? We further trace the hepatic sinusoids. Now these hepatic sinusoids are drained by the hepatic veins, and now one more thing happens, important factor happens over here is the portal vein conveys the 80% of the blood over here into the hepatic, pours it into the hepatic or the liver sinusoids. Now at the same time, even the hepatic artery which carries or which conveys the saturated blood, 20% of the blood saturated blood to the liver, will be poured into the hepatic sinusoid. So there is a mixing of the hepatic blood as well as the portal venous blood in the region of the hepatic sinusoids. So further from over here, so the hepatocytes will take up the further relevant products, metabolic products, and will further process that as a metabolism, process of metabolism in the liver. And after that, the venous blood will be drained with hepatic veins, as right and left hepatic veins ultimately give into the inferior vena cava or drain into the inferior vena cava. So that is how the intrahepatic course of the portal vein can also be described. Apart from being the extrahepatic course, even the intrahepatic course of the portal vein can also be described. So that's one of the important feature related to the course of the portal vein. That's what related to that one. So mind your being in the coming slide, in the next slide will be, if at all, if I'm using as the inferior vena cava, the short form of inferior vena cava has been indicated as IVC, to be representing the inferior vena cava. So that is the extrahepatic, or you can say the intrahepatic course of the portal vein can be mentioned. Whereas now, looking into the parts and relations after being the course, formation and course of the portal vein, now we look into the the portal vein. Now, for better understanding, for better descriptive purpose, the portal vein is divided into three parts. This, as you can recall, this is the portal vein. So it is divided into three parts. Now, that three parts is with relation to the duodenum. So again, more specifically, the first part of the duodenum, with respect to the or in relation to the first part of the duodenum, the portal vein has been divided into three parts, and these three parts are to be as one below the part below the first part of the duodenum is called as the infraduodenal, and the part of the portal vein which is coursing behind the first part of the duodenum to the second part of the retroduodenal part of the portal vein, whereas the part above, which is seen in the right margin of the lesser omentum, is considered to be the supraduodenal part of the portal vein. So this is how the portal vein is divided into the infraduodenal, retroduodenal, and the supraduodenal parts of the portal vein with respect to the first part of the duodenum. So that is how they divided. Whereas now we'll see the relations of these three parts, as you can see over here. So the third part, as I said, supraduodenal part, it is described with respect to the lesser omentum, which I have already told. Now, after knowing the parts, now we'll see what are the relation to these individual parts. So three parts and its own three separate relations when it passes through these it passes through the area below, behind, and superior to the first part of the duodenum. If we take the first part of the portal vein, that is infraduodenal part. Now this is a cross section or a dental section through the abdominal cavity, showing the structure which can be seen from anterior to posterior, from anterior to the posterior aspect. As you can make out, this represents the impression of the liver, and the gallbladder. You can say this will be the first part of the duodenum, and further the pelvis structures in order from before backwards. Now, if we take up the first part, that is the infraduodenal. With respect to this part, now this is the portal vein, what you can see over here, the portal vein, and the posterior most part of the inferior vena cava. Now, mind you again, be it first, second, or the third part, posterior relation to all the three parts, that is the infraduodenal, retroduodenal, supraduodenal part of the portal vein, posterior relation is quite common, that is the inferior vena cava. So what can vary is with respect to the anterior relation to the portal vein with respect to the three different parts of the portal vein. So first we can take now considering this image, now even though this shows it as a head of the pancreas, so this section is slightly to the right of the neck of the pancreas, so that's why I mentioned it as a head of the pancreas. If we consider this to be the neck of the pancreas, now first we'll look at the relation of the infraduodenal part. So the part below the duodenum, so this is the infraduodenal part. So what lies anterior to the portal vein? So mainly the neck of the pancreas, since it is formed behind the neck of the pancreas. So mind you, so remember that. So anterior relation would be the neck of the pancreas. That will be the more specific relation to the anterior relation to the infraduodenal part of the portal vein. So anteriorly, neck of the pancreas. Mind you, so this is like you have to just consider it when you are marking examination secret that not the head, because this is this section of taken from the duodenum relation. That's why this has been written over here. So otherwise, the neck of the pancreas. What lies anterior will be to the infraduodenal part of the portal vein, the neck of the pancreas, and what lies posterior to as a posterior relation to the infraduodenal part of portal vein is the inferior vena cava. So those are relation to the infraduodenal. Now, if it is the second part, which is exactly right behind the first part of the duodenum, this region is the retroduodenal part of the portal vein. So this part. Now, what lies anterior? As you can make out here. Now, obviously, the first part of the duodenum. Slightly behind that, you can see the common bile duct, and further, slightly further behind would be the gastroduodenal artery. So these three are considered to be the anterior relation to the retroduodenal part. This is retroduodenal part. So the first part of the duodenum, common bile duct, and the gastroduodenal artery. So these are the three relations, anterior relation to the retroduodenal. Whereas the posterior relation to the retroduodenal part of the portal vein is the inferior vena cava. That is mistakenly marked as should have been the foramen of the employee foramen or foramen of Winslow, so it has been mistakenly marked. So ignore this part as well. So stay with only the relation to the three parts of the portal wing. First and second we have seen it. The third part, that is the supraduodenal part. So this part is the supraduodenal, since it is above the duodenum, above behind supraduodenal part of the portal. So what lies in front of this one would be the bile duct, the hepatic artery, the chief anti-relation to the supraduodenal part of the portal vein. So what which is behind the posterior is inferior vena. So those are the three relations. I mean, those are the relation to the supraduodenal part of the portal vein. So anterior hepatic artery, bile duct, and posteriorly inferior vena. So those are the parts now. What we learn now is the parts as well as the relations of the portal vein. Now, one of the most important part again, though last but not the least, definitely has to be the tributaries of the portal vein. So what we know that the portal vein receives an extensively from the GI tract, the gallbladder, pancreas, and the spleen. But which specific veins will drain which particular part of the GI tract? So related to that one. What are the tributaries? Now tributaries of portal vein can itself be a 3-marker question. So mind you that. Now the splenic vein, as we already know, the formative veins. Now the portal vein, this is the portal vein. So portal vein is formed like the splenic vein as well as superior mesenteric vein, that we already know that we are already aware of it. Now so in that way, tributary could be formative splenic vein and the superior mesenteric vein are considered to be the two important formative tributaries of the portal vein. Whereas third would be, as you can make out, once the portal vein is formed, you see here on the right side, you can consider this to be the left side of the portal vein. On the right side, you will receive the superior pancreaticoduodenal vein. Superior pancreaticoduodenal vein, draining the pancreas in the duodenal part of the duodenum. So it drains into the portal vein. Whereas, if you stay on the right side, the right branch of the portal vein, if you are already aware that you will receive the cystic vein, that is the gallbladder. From the gallbladder, cystic vein drains into the right branch of the portal vein. So those are the third and the fourth tributary, you can say that. So you magnify it. So the superior pancreaticoduodenal and the cystic vein. Now other veins on the left, if you see on the left side of the portal vein, there are two veins. There is the gastric veins, that is the left and the right gastric vein. The left gastric vein is the longer, so we can make out the left gastric vein, and this is the right gastric vein. Now, what do they receive or what do they drain? The left gastric vein will receive the esophageal tributaries from the lower end of the esophagus or around the cardiac end of the stomach, whereas the right gastric vein, it will receive near the pyloric region of the stomach, that is the pre-pyloric vein of Mayo, as it is also called as. Pre-pyloric vein will drain into the right gastric vein, whereas with these veins, left gastric and right gastric, they ultimately drain into the portal vein. So those are again the other tributaries. As I said again, cystic vein drains into the left of the right branch. Apart from these tributaries, one more important tributary will be around the umbilical region draining areas, that is the para-umbilical vein, which you already seen to be running with the ligamentum teres hepatis. So those are called as para-umbilical vein. So if you see in this image, now this represents a para-umbilical vein that is running with the ligamentum teres hepatis, that is draining into the left branch of the portal vein. So these are ultimately the categorization of the enumeration of the tributaries of the portal vein. So mind you, just to summarize, now the splenic vein and the superior mesenteric or formative veins. Once the portal vein is formed on the right side, you can see superior pancreaticoduodenal. The right branch will receive cystic vein. On the left side, you can see the right gastric vein. This will in turn drain the pre-pyloric vein, whereas the left gastric vein will receive the esophageal branches, the tributaries from the lower end of the esophagus, and ultimately goes into the portal vein. And as well as last but not least, the para-umbilical vein that also drains into the left branch of the portal vein. So these can be separately as we question. So see that we infraduodenal, retroduodenal, and the supraduodenal part, and they have its own individual relations as well. And last what we saw was in the course we even saw about the extrahepatic course and what happens to the portal vein even when it enters into the substance of the liver to be the intrahepatic course of the portal vein. So, and the last but not the least would be the tributaries of the portal vein. Next, what we will see is one more part of this particular topic as well, called as the anastomosis that happens between the portal vein and the systemic veins. So it is also, that's why it is also called as the porto-systemic anastomosis, or otherwise it can also be called as porto-cable. Cable in the sense here, the inferior vena cava. So in relation to that, porto-cable, either can call it as, or it will be called as porto-cable or porto-systemic anastomosis. Now this is a very important part as well, or anatomically it is a very important relevant chapter and topic. Why? Because in the abdominal cavity there are certain sites, as you can see in the diagram, there are certain regions or the sites wherein you can wherein there will be anastomosis between these between the portal vein as well as the inferior vena cava draining channels. So these sites has been, as you can make out in the diagram here, almost about 4 to 5 sites as you can see over here. This is the anterior aspect of the abdominal cavity and the posterior aspect. This being to the diaphragm of the or the upper end of the abdominal cavity and the lower aspect of the abdominal cavity. Now the number one, this would be the umbilical region. So here there is an anastomosis between portal vein channels as well as the vena cava channels. Number two would be around the level of the lower end of the esophagus, or around you can say the cardiac into the stomach. So there will be a rich anastomosis between the portal vein and the inferior vena cava channels. Number three would be around the anal canal. Around the anal canal, there will be a relevant anastomosis between these two channels. And these three sites, number one around the umbilicus, around the esophagus, and around the anal canal. These anastomosis, if there is any hindrance in, or if there is any obstruction to the drainage in these regions, these three regions, umbilical, anal canal, and the esophageal region, they display a very specific signs that we'll see here in the next slide. So very specific signs. Apart from these three, one around the liver, area of the liver, which is a non-peritoneal area of the liver, and even around the one around the liver, and the other one is the retroperitoneal arteries around the kidney as well. There will be anastomosis between the portal vein channels and the vena cava channels. So these are the important sites. Now almost 5 sites wherein there will be communication between the portal vein as well as the inferior vena cava. Now mind your name. So as I said, among the 5, these three are very important. Very important. That is umbilicus, the esophageal, and the anal canal region, because they will display specific signs whatever there is obstruction. We'll see that in the next slide. So that is about like an introductory note. I mentioned that in the portal anastomosis. So this can be a separate 5-marker question, or enumerate the sites of portal-cable anastomosis. So in itself can be asked as a 3-marker question as well, or can be combined with a 10-marker question with the portal vein. So mind you, this is a very important aspect. Now, first we will slightly look into slighter detail about the sites of portal-cable anastomosis. First we will begin with the umbilical region or umbilicus. Now, mind you again, there is slight mismatch between the text part and the numbers which are being marked over in the diagram. So when you are reading, so straight with that, you slightly be aware of this slightly mismatch that has happened over here. Now, out of the 5, the first one, if you take up the umbilical region, around the umbilical region, so again same. So this will be the anterior aspect and the posterior aspect, and on that abdominal wall around the umbilical region over here. Now, around the umbilical region, you can make up what channels will there be anastomosis. As you can around the umbilicus. Now, what would be the portal channel? The portal channel. There should be anastomosis between portal and the vena cava channel. Now, if you take up the portal channel around the umbilicus, it will be para-umbilical vein. We know that para-umbilical vein will drain into the anatomy. As you can make out here, you can see the formation of portal vein. Splenic vein, superior descending vein, led to formation of portal vein, then dividing into left branch and the right branch. Now, keeping that in mind again in the background, the para-umbilical vein, we know that it drains into the left branch of the portal vein. So, para-umbilical vein will form the channel, portal vein channel. Whereas the anterior wall veins of the superior, I can say more specifically superficial venous channel of anterior abdominal wall, they are nothing but the systemic channels that they drain into the inferior vena cava directly. So, they are the vena cava channels, and para-umbilical vein will be the portal channel. So, that is there is now this represents the anastomosis between the portal vein and the systemic veins over here. So, they will. That is forms a very important aspect over in this region. Now, whenever there is now this region, so umbilical region, portal cava anastomosis this side. So, between wall and wall. Whereas, what happens if it does not change? So, this kind of image you can make out here. So, now this is the umbilical region. So, para-umbilical vein running into the left branch. Now, these are the superficial veins related to the anterior abdominal wall, which are considered to be the tributaries of the inferior vena cava. So that is how the anastomosis between portal and the caval anastomosis. If at all, if there is any obstruction related to obstruction, if there is any portal drainage obstruction, suppose for whatever the cause or reason, most important will be the liver cirrhosis. If at all, if there is any alcoholic liver cirrhosis, is considered the most important reason or the cause that will lead to the portal venous blood obstruction. The flow of portal venous blood obstruction, most important cause of the liver cirrhosis, alcoholic liver cirrhosis. Now, if at all cirrhosis happens, there will be a back pressure which is built up in the portal venous channel that leads to obstruction of the portal venous blood flow. So, whenever there is an obstruction to the portal venous blood flow, so obviously being anastomosis, these are systemic veins, that is the caval, the inferior vena cava veins, they dilate and distend. So that gives rise to one important, very important sign around the umbilicus. As you can see, this is the umbilicus. Now these are dilated veins, dilated distended veins around the umbilicus. Now that sign, this page sign is called as caput medusae. Now this is because of obstruction to the portal venous channel, leading to distension, dilatation of the systemic or the inferior vena cava channel. So that is called as caput medusae. Caput means head, medusae means caput medusae is the medusae represents the head. This is relevant with respect to this is related to Greek demigod or maybe something related to that. Now our head, hairs from our head will be like there would be a curve. Now this is a different story altogether. You go to the textbook will give you an idea. So the radiating hairs from our head represent this sign. That is why they relate this sign as the caput medusae around the umbilicus. So that is what happens with respect to obstruction to the portal vein around the umbilicus. That is the first side. If you take up the second side, second anastomotic side, that is at the lower end of the esophagus over here. Now here, which is the vena cava channel and the portal channel. Now the esophageal branches of the left gastric vein. You know that left gastric vein also drains into the portal vein, as you can see here. Esophageal channel draining into the portal vein. Now the systemic veins over here are the esophageal tributaries which drain into the systemic channel that is accessible veins. So there is an anastomosis here. So if there is any obstruction to the portal vein circulation, leads to dilatation, distension of the esophageal tributaries on the lower end of the esophagus. That also gives rise to a very important sign called as again, as I said, most important cause is alcoholic liver cirrhosis for the formation of these signs of the anastomosis to getting dilated up. Now that sign over here abnormal dilatation distension of the venous channel is an varices. So where at the lower end of the esophagus you can make out these varices. This also gives rise to a very important clinical symptom. You can say the blood vomiting, so that particularly called as a hematolysis. So if at all these varices rupture leads to blood vomiting, you might have seen as an alcoholic person suddenly vomits blood. That is because of rupture of the esophageal varices. And the third site would be very important site would be around the anal canal, around the anal region. Now here again there are two, we have to mention about the two sites, like the portal as well as the systemic. Now the portal channel would be the superior rectal vein. Now superior rectal vein is a vein that drains into the indirectly into the splenic vein. Now that again drains into the portal vein. So that's why superior rectal vein is considered to be the portal channel, whereas the systemic veins are middle rectal vein and the inferior rectal vein. That drains into the they drain, as you can make out over here. They drain into this. So as if you go back to this image, now as you can see over here, superior rectal vein is a portal channel, whereas middle rectal and interrectal vein, since they are draining into the internal iliac vein, that is going into the anterior vena cava. So these are systemic channel. This is portal channel. So if there is any obstruction in the portal channel over here, these veins will dilate over here, giving rise to one important bleeding per annum or the anal opening. So that is called the hemorrhoids or the piles, as you might have heard about this term. So that is where you can be put in this image. This is the normal venous channel. Now if there is obstruction, dilated and distended veins giving rise to hemorrhoids like sign. So that is how around the third site. But the fourth site would be around the extraperitoneal. This is with respect to the retroperitoneal organs in the abdominal cavity. That would be more relevant, more related to the duodenum or the ascending or the descending colon. Around those channels or sites, even there we come across the anastomosis between the portal as well as the systemic veins over here. So there, even if at all there is obstruction, that can also be seen as a very close, where you can make out the dilatation of the distension, abdominal dilatation and distension of the systemic veins. Whereas the fifth area would be the pain area of the liver over here. So pain area of the liver. So again, the systemic channels over here are related to the phrenic vein. So related to the phrenic vein or intercostal vein is considered to be the systemic veins, whereas the hepatic vein coming out to the liver is considered the portal channel. So if at all there is an obstruction, cause might be again the most important is cirrhosis. So that leads to again dilatation or the channels, dilatation of the systemic channels around the area of the liver as well. So those are the sites of the portal cable anastomosis. Very important from examination point of view as a 5 marker, or it can be asked as an enumerate. So you have to mention about the channels. So among these 5, most important would be these 3. So these 3, that is around the umbilicus, around the lower end of the esophagus, and the anal canal. So because why important relevant in the sense, they specifically display specific signs. With respect to umbilicus, capital P said, with respect to lower end of the esophagus, esophageal varices, and with respect to the anal canal, that is the hemorrhoids, the form of hemorrhoids. So that's why among these 5 to 6 transport channels, these 3 are very important. See to that you become very much aware of these particular sites. So that almost completes today's topic related to the portal vein and portal and cable anastomosis. Last but not the least is the other aspect related to this channel, that is a portal hypertension. Very commonly seen with respect to person or people suffering from alcoholic liver cirrhosis. Any normal portal venous blood pressure is ranged to be between 5 and 15 millimeters of mercury. If anything which is above 40 millimeters of mercury, that particular condition is called as portal hypertension. What happens whenever there is portal hypertension? Most of it you are already familiar. So one thing would be there will be splenomegaly because of the liver undergoes some cirrhotic change, that is a fibrotic change. So there is an increased back flow pressure in the venous blood, leading to splenomegaly. There will be dissipation of the extravascular comes out of the blood, comes out of the channel and gets collected in the peritoneal cavity, leading to a condition called as ascites. And likewise again, because we increase backflow, there is esophageal varices, hemorrhoids, and capital medicine as a clinical sign. So these are the effects of portal hypertension that can be seen in this particular condition. And second clinical condition would be portal vein thrombosis. This, as the name itself suggests, like any clot formation in the portal vein is called as to be denoted with that particular condition. Causes are very, very wide range of causes to be like anything which leads to inflammatory changes in the pancreas, be it whatever the cause in inflammation, be it viral or traumatic or anything bacterial. Appendicitis can lead to formation of clot in the portal vein. Again, especially in infants, again, quite evident. If at all, if the umbilical cord stump, if it is not taken care of well, so that also can lead to infections again, causing portal vein thrombosis. Other causes would be like abdominal cancer, any abdominal related cancer, contraceptives, oral contraceptives, and abdominal trauma, liver disorders can give rise to the portal vein thrombosis. So those are the upper aspect related to this one. Last but not the least is let's say some of the few of the MCQs as well. Like with respect to this one, like all are correct with respect to clinical features of portal hypertension, except here we have seen splenomegaly, ascites, hemorrhoids, except this one as a normal blood pressure. So normal blood pressure should have been 5 and 15, not 40. Which of the following is the correct statement related to portal vein? The correct statement related to portal vein is like hypertension when the pressure is above about 5 those definitely is not correct. Vein form union of superior descending inferior, again not correct, and both options are definitely not correct. This is to be the more correct answer to the formation and of the portal vein. Whereas as we have seen already, clinical features of portal hypertension to be these medicinal, hemorrhoids, and splenomegaly, not as a very portal liver effusion. Signs of portal anastomosis with clinical sign. As I told you earlier, there are 5 different sites, but most important would be lower end of the esophagus, umbilicus, and anal thing. No doubt about this is the outside side, and these two are the portal sites. But most important displays a very evident clinical sign would be these 3. And as again, this is one more. I mean, last of the see these image-based question. Now, how do I identify what sign? If you already seen, to be tablet, we will say, and this would be like marked area. So, as one would be spleen vein, number 2 would be the spleen mesenteric vein, 3 would be the portal vein formation. So that is how